you just want to do it? All right, I think we're going to go ahead and get started with our session for after lunch. It is my privilege to introduce Dr. Jared Henderson. He is currently the Translational Research Coordinator for the Department of Lymphoma and Myeloma at MD Anderson Cancer Center. He received his PhD in immunology from the MD Anderson Cancer Center UT Health Graduate School of Biomedical Sciences. They have a very short name, right? Under the direction of Dr. Eric Davis, he studied B-cell receptor signaling pathway and its role in lymphoma and developed multiple cellular bioassays to evaluate real-time receptor signaling in living cells. His title today is Don't Sweat Fret, a practical guide to FRET by spectral flow cytometry.
Thank you very much for the organizers giving me the opportunity to do this talk. In the interest of time, I'm just going to go ahead and jump right in. Again, titled Don't Sweat Fret. And the goal of this talk is going to be a kind of 30,000-foot overview, perhaps more of a reframing of what FRET is and how to apply it within the context of spectral cytometry, more than it is a detailed analysis of some data that we've generated. We're going to look at some of that. But again, I want to convince you, hopefully by the end, that FRET is not scary and that any flow cytometrist can do FRET assays with the tools, skills, and knowledge you already have. So jumping right in, for those of you not intimately familiar with FRET, it stands for Forced or Resonant Energy Transfer, and without going into the nitty-gritty details and making it scary, the basic concept is that if you have a fluor, in this case CFP and YFP, which I'll primarily be talking about today, we excite our fluorophore with a laser. In this case, we'll just call it the 405 violet laser. And we have that fluorophore accept the energy and then transfer that out with photons that we can read on our instrument according to the emission spectra of that fluor. In the case of FRET, if we have an suitable FRET pair, in this case CFP and YFP, we take that energy and instead of our donor fluor, CFP, emitting light that we can detect, that energy is then transferred to our acceptor and given off at its corresponding wavelength instead. So if we take a look down here, again, this is mostly to read the distance between two fluors. If we have a FRET pair that has significant distance between the fluors, we excite our donor fluor, and then we are going to be able to read the emission that comes out at that appropriate wavelength. But if our FRET reporter system has both fluors in sufficient proximity, we'll be able to read a corresponding downshift in the emission spectra generated by our donor fluor and an increase in the relative emission of our acceptor fluor, again, based on spatial proximity. Again, there are a lot more facets of FRET than I'm going to get into today, but that's basically where we are. And common FRET uses in flow cytometry, there have been several groups that have done a protein-protein interaction assay. That's not really what I'm going to focus on today, because we have more experience with these FRET-based biosensors that function similar to what is described here, where we can do signal pathway monitoring and some small molecule and chemical sensors. So the reporters that I'll focus on are kinase reporters, and specifically I'll talk about our AKT reporter that we've characterized, I think, fairly well, where we have a single transcript FRET reporter that contains both the donor and acceptor fluors and a FHA1, which is just a phosphoamino binding domain. And then in the case of our AKT reporter, a FOXO1 domain that is phosphorylated by AKT. So you can see that the basic premise here is that when the FOXO1 sequence is not phosphorylated, these two fluors are allowed to kind of rotate around in free space. They are not in close proximity, so we get low or no FRET. However, if AKT is active, it phosphorylates its target, FOXO1 peptide. The FHA1 domain binds to that phosphorylated residue and brings our proteins into close proximity, and we get a FRET signal out of this single molecule reporter. And that target peptide, as we've shown, can be changed specificity to other kinases. So, pick your favorite kinase. You can adapt it with just changing a few sequences, a few amino acids out. And the reason we got into this, just for a little bit of context, is we were interested in different mutations, signaling mechanisms, drugs in the context of lymphoma for the BCR signaling pathway. And downstream of that is AKT signaling. So we were looking for a quick and easy way to be able to determine the net effect of any perturbation in the BCR signaling pathway on AKT. So hence FRET. When we got into this, I'll do this the next couple of slides as kind of a montage instead of actually digging through the data. We started with microscopy and got really great data using this reporter. And you can see here, again, great tightness, great data, but each one of these dots is a single cell, and this took weeks to collect even after the system had been built. It's incredibly labor intensive. So we then moved to conventional cytometry that uses essentially the same methods that you would use in microscopy, and we're able to expand that out to the cell lines that you see here, a lot more data. And instead of single cells, we're doing thousands or tens of thousands of cells. And that worked great and was much less painful to get. But that usability increase comes with some pretty big asterisks as far as flow cytometry is concerned. I won't go through all of the stuff that you need to actually make this happen and make it mathematically rigorous to get a FRET efficiency rating. But it was sufficiently rigorous that we ended up making a package that we've published in R to automate some of that downstream math so for cytometrists to be able to use it. And again, we'll just skip over this, but I'll just say, even with these improvements in using flow instead of microscopy, you're talking about hours of post facto analysis and running a lot of individual controls, not just for the assay, but every single time you run the assay, you're doing all of this controlling and then after the fact analysis. So I'm not going to acknowledge the clear benefits that both of the other two methods have. What I'll say is that the problems from a user perspective are that you need custom controls, which you'll have to make, design, implement, tweak, so on and so forth. And more importantly, using FRET requires a set of knowledge and skills that are not typically possessed by your average flow cytometry user. And so basically what happens is if you want to ask a FRET question, you need to familiarize yourself with that literature, build a bunch of skills, and you don't know whether your assay is actually going to result in the data that you need downstream anyway. So it's a pretty high barrier to entry. So the dream that we had, if you'll permit me to say that, is that we want a FRET assay that works just like any other flow assay that you're prepared to run in the lab. Similar kinds of controls, analysis by regular flow cytometry analysis, FlowJo, FCS Express, pick your favorite analysis tool, and get that data in real time on the instrument, and if we're dreaming, the ability to sort based on that FRET signal as opposed to taking your cells off, doing an analysis and going, hey, it would be great if I could grab those cells. And the answer here is spectral or full spectrum cytometry. So to mentally reframe FRET, the assay, how we're actually going to get there, first, normal FRET controls won't work. I didn't go into this in detail due to time, but in general, when you run a FRET assay, you need 4 controls. You need to be able to identify the spectrum given off by your donor and acceptor fluors separately. Then you need to look at what FRET looks like and what low FRET looks like with the presence of both of these fluors. You need to calculate a bunch of crosstalk coefficients, et cetera, et cetera. It's pretty onerous. And if you look here at the spectral signatures given off by these fluors, you can see that even for spectral flow, those are pretty similar. And your instrument is going to have a hard time picking apart which component comes from where. And so basically the answer is that you don't need to do any of this. We're already all doing FRET all the time. You just need to get a reframing of that. Our tandem dyes, I've chosen PE Sci5 here as just a random example, is not a fluor. It is just a highly efficient FRET pair that we've been able to harness to stain our cells. So if you're planning a panel, you would not treat PE Sci5 as PE and Sci5 and as a FRET pair. You would just say, hey, spectral unmixing software, here is a fluorophore. Go find that signature and move along with your day. And so I just point out that these are not the same. And so we shouldn't treat this the same in a FRET assay. So again, what we're looking for is FRET pseudofluors, not individual fluors. The key points, and it was great to follow Diana talking about autofluorescence this way. Your spectral and mixing software doesn't care what you call something. It cares about the spectral signature that's coming out of it. So there's no reason to think of something as a single color you need to think of it as a single spectrum. And that works for autofluorescence, and it also works with FRET. And for me, this was a challenge, is to ignore the intuition that a fluorescent protein must be a single thing. If your single color proteins are always occurring in pairs or are occurring with a spectral signature that comes out as a single readout, a single color control can have multiple fluors in it as far as the spectral unmixing algorithm goes. So show the algorithm what you want, and it will try to unmix just that. So reframing this, we no longer need the individual contribution of in this case, our donor acceptor, cerulean 3 and Venus, because our FRET output already encapsulates the contributions from either of those individual fluors together as a single spectral signature. And in this reporter, we only have low and high FRET states, not C3 or CPV alone. So we're going to move on and ask, what does that look like in practice on, in this case, a 4-laser Cytec Aurora? First, designate low FRET and high FRET spectra as pseudofluors. This could have been Alexafluor 647, but in this case, I literally just called it low FRET and high FRET and fed in as my reference control the reporter constructs that we've built for low and high FRET. And you can see on these plots here that the spectra are fairly similar between these two, except that in V7, which is our traditional channel-based FRET channel, that there is a significant difference, or I guess relatively significant difference, between the intensity in that FRET channel where we're seeing that FRET emission off of our acceptor protein. And I also just want to point out that a big reason this works is the autofluorescence extraction. You can see in our autofluorescence down here, our unstained control, just to make my life complicated, V7 is our FRET channel. It is also the peak emission of autofluorescence for our lymphocytes. So, you know, a lot of the work that we were having to do computationally was to get rid of that autofluorescence, and Diana demonstrated quite nicely how effective that is in cleaning up signal. And so once we do that, you click the unmix button and you have FRET being read out as any other fluor that you would have put in your panel, period. Because the algorithm is already doing a lot of that back-end computation for you, and it's doing it in real time. So what does that look like in practice? So over here, we have the traditional FRET real-time output where we have channel ratios between the acceptor and donor channels. This is both on the Aurora and the Fortessa. It's not instrument specific, it's method specific. But when we do the unmixing, where we just choose to make an arbitrary fluor, you can see that these are actually the same samples on the Aurora. These are identical events paired together, just showing you a different plot. We now have low FRET and high FRET on our axes as we would with other fluors, and we can read the changes in FRET efficiency just as more or less a linear output of FRET efficiency across these plots. And I don't know about the people in this room, but I don't feel great about gating these separate populations, but I feel really good about being able to discriminate these populations and use them either in a downstream analysis or for a potential sort, as we'll get to. And I won't go into much of the data here just on account of time, but I will point out that you don't really lose much as far as fidelity of the assay in the 3-cube more rigorous mathematical method versus SU FRET. This is a live kinetic assay. It gives us the same readout. But what you do gain in SU FRET as opposed to these channel-based methods is essentially rendering spectral overlap irrelevant. We see that in the channel method and in our E-FRET method, 3-cube method, we stain the BCR with different anti-BCR antibodies or anti-IgM antibodies, and we can see that the presence of dialite, which if we look at our channel here in gray, dialite 488 shows up right in the middle of that FRET detector, and it throws off our calculations. But SU FRET does not care because it's able to fully pull out that 488 signal, separate it from FRET, and move along. We can also, as flow cytometrists, I would be remiss if I was talking about spectral flow and not say something about high parameter flow cytometry. We can still do FRET even with a fully stained human tonsillar B cell panel. This isn't the craziest panel, it's a proof of concept. We can do all of this staining. We still get good fret resolution. So if we look at fully unstained versus fully stained cells and overlay those contour plots of fret channels, we get no sacrifice in the resolution of the fret, even with the presence of all of these other fluors. And again, as I pointed out, kind of our dream here was to be able to use fret as a real-time readout and be able to sort cells based on the status of fret after, say, a CRISPR screen or drug screening. And I'm happy to say that after some time, we've been able to accomplish that in real life with an Aurora CS with the MD Anderson Off Campus flow cytometry course, so that's been very exciting. So as far as the practical piece, what does this actually look like? What do you need to do as a user? First, define your fret states. And again, I think the meat of what I'm saying here is that you don't need to treat fret as a special thing. Treat it as a fluorophore or as a spectral signature. What are your fret states? If you have this biosensor, you only have a high fret and a low fret state. You only need 2 spectra, not 4, because you're destroying your ability to detect that signal by giving the algorithm more stuff to try to piece apart. If you're doing something perhaps more complicated, like a PPI fret, protein-protein interaction, we've got some experience with this. In this case, you wouldn't have a low fret and a high fret because you're not dealing with a stoichiometrically balanced fret sensor. You will actually have spectra from your donor and your acceptor, and then you'll also have a fret state. So you have 3 spectral reference controls that you'll need to have there. And again, you'll have to do some thinking about your biology to determine the strength of that signal, which is our next and final piece. What is the dynamic range of the fret reporter that you're using? Hopefully, you're going and grabbing something that's been well characterized and published from AdGene or somewhere. You're throwing that in your cell. And you can use, as I've gotten here, fret high and low controls that are just artificial constructs, where we've got a large dynamic range of, say, 10 to 40% of our real-life biosensor. But if you have a lower dynamic range, or you want to kind of narrow that range so that you can get better gating or some increase of resolution, I don't really have time to get into that, you can actually use your fret reporter instead of building custom controls. And we do this all the time. I think this is kind of the scariest thing about fret is we need all these extra controls. If your reporter behaves well, there is no reason that I couldn't, after running an assay, say, well, this green population is my fret high, and this blue population is my fret low, and I'm going to use my unmixing algorithm to just make those our reference spectra. And now you've essentially expanded out that distance on your orthogonal plot. You may not get increasing resolution, but you can use it. So again, you may be able to do a fret assay without bothering with all of these fancy controls, again, with some caveats. So with that, I probably just already said most of this, so we'll just say for unmixing, think in terms of the total spectra, not of individual fluors. It's just a moderate reconfiguration of expectation there, and determine whether you even need custom controls or you can use what you already have in a well-characterized reporter system. And then record, unmix, and enjoy that data. And again, fret isn't fancy. It's just like any other fluor with some thought. With that, I want to thank my collaborators here, and I'll take any questions maybe, and I'll leave us with a little video. This is actual real-time kinetic assay on the instrument, sped up 8 times, just sitting there watching it after a BCR stimulation. Sorry, go ahead.
Thank you so much, Jared. That's a fantastic work. We read your manuscript and it's pretty impressive. Well, you have all the perfect controls, right, to prove that it's working. So thank you for that one. And just curiosity, have you checked what's the similarity index between your low and high fret populations? No?
No. And
they're very close, right?
They're extremely close. And we've actually, so I've mostly talked about our AKT reporter here. We've got several biosensors. We've got AKT, PKA, and calcium flux reporter, which is also beautiful. but they're such different systems as far as what the dynamic range is and what the similarity is. And I think to completely avoid your question, the short answer is it doesn't seem to matter all that much. It obviously matters kind of writ large, but because you were just making a comparison between two very similar things and you're just letting the algorithm say, is there a little more of this versus a little more of that? It works. It just works. And that's kind of the beauty of the system. Even when we had a hard time parsing it out using this very complicated cell by cell math and crosstalk coefficients, it wasn't as clean as just throwing it into the software and going, even when the differences were pretty minimal. Mm-hmm. Right. And is there another? I
Hey, thanks for that. It was a really nice talk. You just mentioned some other sensors. That was the first thing I was going to ask you about. So are you starting a company? Are you building new sensors? So that's one question. And then the other one was, have you introduced these reporters into a mouse so that you might be able to do this in vivo or at least ex vivo on normal cells?
Right. So if anyone has an absurd amount of money they're willing to throw at me, I'm really open to that. No, yeah, we're using a lot of different kinds of sensors, but essentially this what we found is that this single molecule biosensor, where you don't have to worry about stoichiometric balance between the floors, works just beautifully in the system. We could have even designed a little off multiple fret reporters, fill in the blank. It's just an easy to use system. Now, as far as the putting this in a mouse, this is not a particularly challenging construct to get in. If you can put GFP luciferase in a mouse cell in a particular lineage, you can put a fret biosensor in there. We don't have expertise in the mouse component, but again, there's no reason that you can't transduce cells like this, pull them out of the mouse and get, say, AKT, PKA, some reporters that we already characterized here, out of a germinal center, for example. There's just no reason you shouldn't be able to do
it. Okay. They definitely turned the gain up on this thing. So can you say a little bit more about, okay. Can you say a little bit more about what you would have to do when you do have concerns about stoichiometric balance in order to do protein-protein interaction work?
Yeah, I can to an extent. We've done a little bit of work on this. Most of that work we did when we were still using the 3-cube fret method. As far as the spectral method, the key is that you can't show the software spectra that you'll never achieve as in the cell. So as I mentioned, you need a donor and an acceptor floor spectral signature that you're identifying. But, and I don't have this data here, we actually used, looking for these PPI, transient PPI interactions between, say, CD79A and SYK kinase. I actually used the low fret control that has a rigid spacer. So the fret that comes out of that is extremely low. And so if you use that as your spectral unmixing fret signal, it still works, because what you're going to get is relative to the whole signal from the cell, you're going to get relatively less fret relative to the amount of total reporter available. So that was kind of a winding, meandering answer. It can be done. We've seen it. We can do kinetic assays. We can do time point assays using the PPI, but you have to be a lot more deliberate about your controls and probably do some empirical testing like you would for, again, another flow panel. All right, thank you.
Right. All right. So our next speaker, which many of us know and feel favorable upon, is Nico. So I actually have a little something here. It's so Nico has over 10 years of experience in flow cytometry, as we all know. He currently is a senior application scientist, multi-omics specialist at BD Sciences. But I know him better from when he was the SRL manager at Baylor Immunology up in Dallas, and known him since then as he's been collaborator in FlowTex since then. He is additionally in 2021, Nico moved to industry and joined the BD FlowJo team. I will say now I'm getting a lot better responses from FlowJo, even though I had great ones before. Now I can actually call his cell phone, so it works out pretty well. Anyways, but he was also previously in the ABRF research group, and as well as a recognized member of ISAC. And please, Nico, come and give us your talk. Oh. One last thing. This is driving me crazy.
Put it on the other side. There's a FlowJo logo on the left side. Nobody cares about the FlowJo logo. All right. Welcome, everyone. So thank you, Joel, for the introduction. So my talk will be shorter than what's supposed to be at the beginning. But I'm sure it's going to raise a lot of questions. And so we're going to use the time. We already had 2 hours of questions and talk about compensation last night at the speaker dinner. So I'm sure we can keep going on that topic a little bit longer. So today I'm going to present AutoSpill. It's an algorithm to ease compensation and remove autofluorescence. So just to be all on the same page, I'm going to start with a glossary of the different names. So we call the same thing, the same name for the thing. We maybe have a different name, sometimes depending on the company, and especially marketing people that try to call things differently to make it fancier or more sellable. Then we're going to talk about traditional compensation, the one that everybody knows, and I'm going to present the differences with AutoSpill. So first, fluorescence. What's fluorescence? So it's the capacity of an absorption and emission of light in a short period of time by a molecule. So like Jared mentioned, so you have an energy of light, the molecule absorbs the light, and you go in a different state of excitation, and the molecule comes back, and when it's coming back to the ground state, emits some light. The fluorescent dye, so it's a molecule that's fluorescence, and it can be quantified for detecting a phenomenon. So phenomenon for us is usually antibody marker. And those fluorescence dye can be used in many different techniques. And of course, we here in the flow cytometry, but can be microscopy or fish. So the detection of dye doesn't matter if it's full spectrum or traditional cytometry. You have an excitation point and you have an emission for every dye. If anything at big P psi 5, like Jared. So you have always a primary detector, and for the primary detector, usually the primary detector is the one where you have the maxima, so the maximum excitation, maximal emission of light, and all the other detector are called secondary detector. And the spillover, it's whatever the emission is coming just getting into those secondary detectors. So the spillover of each dye into each detector is calculated, and the value is arranged into a matrix. So we get a spillover matrix here. And for traditional spillover value are shown as percentage relative to the primary detector. So for the unmixing, sample with a full complement dye are called fully stained. We have a signature for all those dye for the sample, and basically unmixing is knowing for each dye how much it contributes to that signature for each detector. And I think Diana present that really well. All right, and so composition is just a particular unmixing algorithm. It's not anything different. It's just a specific case of unmixing. And basically, you invert the spillover matrix inhibited by the data. So the word composition comes from the idea that you're fixing the spillover. If spillover is a problem on your data, but this is only an algorithm for unmixing when you have one dye per detector, or the traditional flow cytometry case. So how do you estimate the spillover? Well, you just run a single color control or monochrome to the instrument, and you tell the instrument, that's my FITI, or that's my PSI5, and try to see what's the signature of where my spillover goes on every other detector. So to do good composition control, you have to follow 4 rules, and they've been established by Mario there. So the Super Mario rule, as I call them. So the first one is make sure that you match the exact fluorochrome that you use for your composition. So if you have GFP in your sample, of course, you need to use GFP for your composition. Also, the second rule is to make sure your background fluorescence should be the same for your positive and negative control. And doesn't mean for every color, but just for the same color. So for your control of GFP, it should be the same control. The control need to be at least as bright or brighter than any sample of the composition the applier on. And the fourth rule is to make sure that your control follow the same treatment that your sample has. That's particularly true for fixation and permeabilization. So at the end, after you run all your control, in the traditional flow cytometry, you get a spillover matrix, you do a composition matrix, and you get the percentage for every secondary. For spectral unmixing, it's kind of the same idea. You get a spectral matrix instead of a spillover matrix, and you do a spectral unmixing matrix. Now let's talk a little bit of autofluorescence. I'm just going to repeat a lot about what Diana just said, but autofluorescence come from the cellular from the organelle, so mitochondria, lysosome, a variety of intracellular compounds that contribute into the fluorescence. The larger your cells are, usually the more autofluorescence you get. And beside the cells, also the treatment or the condition of your experiment also influence autofluorescence on your cells. So incubation with buffer, presence of dead cells also will influence your autofluorescence. Presence of residual hemoglobin, also if you didn't lyse properly your red blood cells in your sample, and of course the treatment or excessive concentration of paraformaldehyde. So in this circumstance, in addition of proper fluorochrome assignment of panel designs, where you're going to try to remove, use fluorochrome that far from the autofluorescence for markers that are really important in the question, it also can be beneficial to remove autofluorescence using a mathematical calculation. So let's talk a little bit about traditional composition. So traditional composition is the one that you find everywhere on every software. And basically start with you doing a cleanup gate around your cell of interest to know the consistent autofluorescence. And you have to be careful by doing this cleanup gate. This cleanup gate need to be tied around your population. So if you use PBMC, you're not going to do a gate on all your PBMC, but maybe just your lymphocyte if you're looking at a CD3 marker, for example. So, and because, as you can see, the background of autofluorescence really differ between the different cell population. Then you're going to create a negative and positive gate, and we're going to with those gates, we're gonna determine the median fluorescence intensity for the negative and positive population. Then we're gonna calculate the differences for the secondary detector, and we're gonna remove that spillover. So in this example, a classic FITCP example, in the removal of that spillover, we just result by this mathematical calculation, which is 16 minus 2 divided by 100 and minus 2 multiplied by 100 to get a percentage, which is 15.05% compensation. And then we do that for every secondary detector and get the full spillover matrix, or compensation matrix. At the end, if the compensation is done properly, the signal for the secondary detector should be the same for the negative population and the positive population. So we just do positive and negative event for each single stain control, and it's easy, right? But not so easy, because you have some difficulty with low frequency expression of marker, or low antigen density expression, or autofluorescence. And in this example, for example, we had low frequency expression of this TCR in FITC, and we had only 0.72%, which was not enough to calculate a composition. So we look at the 2D on the dot plot, and we see our median fluorescence intensity is right there. And we're like, okay, so a lot of users have the tendency of moving the gate and moving to the left to get the 2% to be able to calculate the composition. So, but moving that gate changed your MFI. Your MFI now dropped from here, which was a little bit over 10 to the third, to not even 10 to the second. That means that your positive control now cannot be more positive than 10 to the second, which is not going to be the case if you want to have composition. So now we're going to switch to AutoSpill. So AutoSpill is an algorithm that's been published in Nature. And here's the title of the paper if you want to read it. A method for calculating spillover coefficient in high parameter flow cytometry. It's coming from Dr. Adriaan's Liston lab in Belgium. And it's available also on their website. All right, so how AutoSpill works. So first, AutoSpill, you don't have to do gate. You don't have to do any positive or negative gate to determine the spillover. What it's going to do is going to use a linear fit on your data. And basically, it's going to try to find that slope of your composition, taking in consideration that doesn't matter if the intensity of your positive sample is still a percentage, still the same percentage that's going to be removed for the composition. And it's going to try to bring that slope back to have a 0% slope. So you still need to use a single step control for your composition. And your composition, as I mentioned, will be used by calculating the slope. And the slope is going to be, the way we're going to calculate the slope, we're going to use a robust linear regression line to calculate that composition control. And the version is exactly the same as for traditional control, so traditional composition, sorry. And the good thing is you don't have to do positive or negative get needed, so it makes it easier for do flow cytometry. Another advantage also is you don't need to have particularly good control to run your composition. So this type of control that in the past was not optimal to run composition because you didn't have a clear population. Now with this type of control, it's pretty easy to get the slope and to calculate the composition. It's even better than having beads, for example, to calculate composition. So I've been talking about a robust linear regression. I just want to go a little bit deeper into that. So what robust linear regression means is we're going to give a lower weight to events that are further away from the estimate regression line. So the event that here, for example, in this drawing, will have way less weight than every event that were like close to that line of composition. So in a real word, so that means here in this event, all those events maybe right there that not really go into the slope will have a less weight. Also, we talk about linear, but when we look at the P exponential, we don't see any thing that's really look like a line. But if we switch to a linear scale, we can see that composition is really linear. So easy to find the line of the composition and bring that line to have a 0% slope. So after the first calculation of AutoSpill, the good advantage of that algorithm is going to keep iterate to improve this linear regression. So it's going to try to do it first. It's going to plot it back again, try to see if you can see any slope again, and it's going to try to it's going to correct it again until it doesn't see any slope anymore, and it's where until convergence, basically until the slope is at 0 and there's nothing to correct anymore. Also, the good thing with AutoSpill is you can remove autofluorescence. So autofluorescence is the spectrum of cells, and as the mentioned by a speaker before me mentioned, so it's just a parameter that we can open on the machine. We can have a signal for that autofluorescence, and with that signal, we can remove that autofluorescence with the AutoSpill algorithm. So you just have to choose a parameter with high autofluorescence signal. Usually violet or UV laser excitation, a maxima around 530, between up to 600 emission, and you just need to have an open channel available in that range. Also, for the cleanup gauge, make sure, because the autofluorescence depends on the signal, make sure that you're on the cells that have a high autofluorescence signal as well. All right, so but composition is done, but we forgot about spreading. Spreading, when the tragedy SSM calculation, use a negative and a positive population, but here we don't have anything. So how can we do to calculate the spread, the SSM? So we found a solution creating this auto-spread algorithm. So we partition the data into quantile. We estimate the fluorescence in primary detector. Then after we estimate standard deviation in each secondary detector, and we calculate a linear fit to determine the standard deviation at 0. So this line, we're just going to bring it to 0, as like that. Then we adjust the data, we calculate the linear fit to determine the spillover spreading coefficient. And at the end, for most data set, the auto-spread produce SSM really close, almost identical to the traditional algorithm to determine the SSM. So what's the advantage of auto-spread, and why we really recommend you to switch using those algorithms instead of traditional composition? First, it's way less demanding because you don't have to adjust gates, do gates. So you just bring your, use your composition control, run the algorithm. You just have to make sure your cleanup gate is at the right place and nothing else. Also, it should not require any manual tuning into the spillover matrix. So because the algorithm is going to automatically iterate the calculation, it's going to improve the composition until it thinks the composition is optimal. And even if there was a big discussion yesterday, even if manual tuning is not recommended in the field, we have a lot of users still doing it, and something that we try to avoid doing it, and we're not recommending anymore to do it. Also, the autofluorescence can be removed with that channel, which is a big plus that just with traditional composition, but you cannot remove autofluorescence. So, of course, with advantages come limitation, and I want to be really clear about the limitation of AutoSpill. So first is to perform a linear regression, and you need to be able to do that. If you're not able to do a linear regression, AutoSpill will not work. So it's most efficiently to do a linear regression, on cells than on beads, because beads are so condensed, so you're just going to have a point of positive cells, of positive beads, a point of negative cells. So the line is going to be a little bit harder to define. Then sometimes with the fine tuning, the adjustment, some data may fail to converge into the solution with a lower composition error. So even if we don't recommend to judge a composition, sometimes with AutoSpill, you're not going to get the best composition anyway. And also, of course, the autofluorescence should not be estimated on beads, but should be estimated on the cells that produce the most autofluorescence. So here's some example of the improvement of AutoSpill, and this one's only on composition. So here's the traditional composition on Treg population, and that clients were losing this Treg population. And when we backgating, we saw that the reason it was losing his Treg population is this CD19 TCR beta, all his Treg population were out of the gate. And that was the result of a composition issue on the CD19, CD25. So we didn't trick it manually. We just ran AutoSpill with the same composition control. We didn't change the composition. We ran exactly the same tubes as composition control, but into the AutoSpill algorithm. And by running into the AutoSpill algorithm, we were able to restore the Treg population automatically. And also, the AutoSpill improvement is on the autofluorescence. So those are high autofluorescence GFP cells, and I cannot say too much in there about that, sorry. But was the composition without autofluorescence removal, where we had only a 9.21% of GFP positive. But with the removal, first of all, the negative data looks way better, and we were able to get a 20% GFP positive cell. So a big improvement in separation between the negative and the dim population on GFP. So, of course, because I'm FlowJo, we implement that into FlowJo. So if you have FlowJo 10.7 software, you have the AutoSpill composition available now. You just click on composition, and you can pick either traditional or AutoSpill composition now if you want. If you don't have FlowJo, I don't know why would you not, but if you don't, you can do it automatically on the AutoSpill website. So you just go on the website, put your composition control into it. It will create the spectral matrix for the composition matrix for you, and as a CSV, and you can import that in any other software. Also, on top of AutoSpill, you have also you can do also spectral composition. And spectral composition, yes, you have full spectral composition. Cytec and BD also have full spectral composition, but any instrument can do spectral composition. You just need to leave open all your channel on your instrument, and it will allow you to have a better signature of autofluorescence on your signal, and be able to remove a better to have a better composition. So highly recommend it to have all your detector open if you're not even using all of them. And the last option that we have available is to optimize weight. When you do a spectral, a big issue is the spread of the data, and we have an algorithm to optimize the weight, which is gonna decrease the SSM for your data. So basically what's gonna do is we're gonna try to find we're gonna put some weight on different detector, and we're gonna see how it's gonna affect the SSM. And when we see that the SSM is the lowest, we're gonna keep that weight, and we're gonna run that into your data. Because it's really computer intensive, just be aware that if you check that box into a FlowJo, just be ready to take a coffee break or lunch break before getting the result. And thank you for your time, and happy to hear any question.
Yeah.
I don't have 2 hours. Just say that.
Exactly. Exactly. So I'll just keep it short because I do have a lot of questions. but just no, just out of curiosity, that example that you gave with the traditional compensation, with the Tregs that didn't really work very well, and then when you apply the AutoSpill, you actually rescue the Treg population. So I wonder, why didn't it work with the traditional? As long as you have the right controls, etc. I didn't understand why it didn't work.
Because AutoSpill, because you calculate the slope, you're less dependent of the intensity. So even if your sample is brighter, which in theory is not a good condition for your composition, because you calculate the slope. As long as you have the slope, even if you're brighter, your slope is still correct. So your calculation, the accurate of your calculation of your slope is better than just removing the spillover.
Okay, I see. I see. So you basically you didn't have the exact right controls, didn't work, but without those.
Exactly.
Okay.
You're a little bit less flexible on the control.
Okay. Thank you.
I think the like autofluorescent removal is great. And I know Cytec has come out with the Aurora sorter, but since now FlowJo is part of BD, does BD have any thoughts on adding autofluorescence and removing to, you know, the older sorters like the Arias and stuff, so that we can remove the autofluorescence and sort our GFP better? Because you know, everyone sorts GFP and they're always autofluorescent, and it would be really nice if we could see 20% instead of 5% because of autofluorescence.
So you can remove autofluorescence on Diva if it's a spectral instrument on
Yeah,
Diva.
I'm talking about like
on
the older ones.
on traditional. I don't know that answer. You know, but we still come a split between FlowJo and the rest of BD, so I don't have that answer on the instrument.
Okay, thanks.
But because you can get the spectral, if you run your composition on Diva, you probably can export your matrix and put it in Diva after from FlowJo. So you may be able to do it. Yeah.
So you said that this new algorithm works best if you have the negative control in the same tube. So would you basically spike your sample with unstained cells to get the negatives in there, or how would you do that in practice?
Sorry, Billy. Can you repeat?
That to get negative cells in your single stain control, assuming depending, but in some lineage markers or something, you might have basically 100% positives or something. So would you spike that control with unstained cells to get the negatives in there, or concatenate two different files afterwards? Or
yeah, I think with cells it's very rare when you have 100% of positive, but yeah, in the case you have 100% of positive, yeah, you will have to find a way. You can always use beads. It's not because you use AutoSpill that you cannot use beads as composition. It just will be, I'm not gonna say less accurate, because basically it's just gonna give you exactly the same number as traditional composition. It's not gonna be an improvement, but it's just gonna give you that number. Compared to that with AutoSpill, you're gonna have an improvement of composition.
Thanks, Nico. Great talk. I wanted to ask about the optimizing weights for unmix. Does that take into account what your panel is, or is it just running through a process of what happens if I set the weights to this? Like, how does that work?
So it's just gonna, so the weight is at 100% for all detector at the beginning, and it's gonna move the weight for each detector differently, and look at how your SSM is gonna vary between when it change each value. So if it's gonna move a detector at 60%, how's the spread? Is it gonna be bigger or smaller? It's gonna do that for every detector. And when you find the optimal, that's where it's gonna set the weight for every detector.
I wonder if you could fine tune that depending on what your panel is, you know, because I can think of a lot of floors where maybe you could see where you would have more weight on some channels than others.
Yeah, Rena is automatic. You don't have you cannot fine tune it manually, but yeah, it will be interesting to look. Yeah. Yeah.
Do we have any other questions? All right, I think this means we're going to take a coffee break until our afternoon session. If let's see here, we have a time 2:15. If we could reconvene at 2:15, and see you then.
That would help. Okay, welcome back. Hope everybody is well caffeinated and still awake. Joel was just mentioning we're doing a raffle at the end of this session. And for everybody who's in person, you get to drop your badges in a box. If you registered online and you are still here online, the great news is you can still win some gift cards, but you do have to be here to claim them. And you do have to have the chat enabled, which means you need to be logged into a Google account as well. So if we spin the little wheel and your name comes up, you have to reply in the chat. So if you're not logged in, log in. Okay, let's get ready for the next talk. Our next speaker, our first speaker, this is a cell sorting session. You know her, you love her. Her name's Clise-Dwyer. She's the core director here at MD Anderson at the Advanced Cytometry and Cell Sorting Facility at South Campus. I'll give you her real information, which is that she earned her PhD at the University of Wisconsin-Madison, where she studied B lymphocyte deficiency, and then did a postdoctoral fellowship at the Trudeau Institute in Saranac Lake, New York, where she studied CD4 T cell function and aging. She's directed UT-MD Anderson's Advanced Cytometry and Sorting Facility since 2008. She's an associate professor in the Department of Hematopoietic Biology and Malignancy and Stem Cell Transplantation, and has been a professional cytometrist for over 20 years. Her research at MD Anderson focuses on single-cell analyses in leukemia. She's a past president of FlowTex and has been a member of the FlowTex organizing committee since it was formed in 2007. And I think what everybody in this room, if you probably do actually know her, and probably a lot of people online too, and it's because she's very impactful in the community. She's been very impactful in my scientific journey as well. And so I'm excited that she's talking, and I hope you guys are too. So let's give it up for Karen.
Good afternoon. So my talk today is intended mostly for an audience who is not that familiar with cell sorting. So some of you may just not want to pay attention for the next few minutes. For cytometrists, I think a lot of people do flow analysis and don't necessarily do sorting. And for researchers who don't specialize in flow, I think there's a lot of people who come to the core labs to do cell sorting and just treat it as a black box. And I would like to have people improve their experiments by understanding a little bit better how the technology works. So with no further ado, I'm going to start my talk, hopefully. There we go. Okay, so the outline of my talk is a few slides about how sorters work and how they're different from analyzers. And then I'll have a lot of FAQ, which are questions that we get asked by every customer who comes into the lab and wants to sort cells. I want to stress how important sample quality is when you bring a sample to the core for cell sorting. And then we're going to look at some best practices and some of the cool things you can do with a cell sorter. So just a quick overview of the hardware for cell sorting. It's a pressurized system where you're essentially pulling or pushing sheath fluid or PBS from a pressurized tank down through a nozzle and flow cell. In the flow cell, you're injecting the sample, and you're using hydrodynamic focusing within the flow cell to push those cells into single file. And then there is a small aperture through which those cells pass. That is called the nozzle. And the nozzle is usually a small aperture in the range of 70 to 130, sometimes a little smaller, sometimes a little bit bigger microns. Then, as with any flow cytometer, you're going to have laser intercepts and detectors. I'm not going to talk too much about that. Mostly, I'm going to focus on the mechanics of the stream and drop creation, as well as how we're going to get those drops once they're charged into the collection tubes that everybody wants. Everything that doesn't get sorted comes out through a waste catch and goes into a waste tank. So it's pretty important to be focused on how the sorter works so that your cells of interest end up in your tubes and not in the waste tank. Before any talk about cell sorting, we should talk a little bit about the biosafety of cell sorters. Cell sorters are a special hazard for biosafety. There's an elevated risk because sorters can produce large visible clouds of aerosols that are sized pretty well to fit into go in for deep lung to go deep into your lung tissue. Anyway, so Those particles will vary between about 50 and 500 nanometers. The 50 nanometer particles can stay in the air for a long period of time. So there needs to be protocols to assure that your aerosol containment has been validated. Your sorter should be placed in a biosafety cabinet in a room without additional equipment that should be used at the same time. You need to have negative air pressure, basically a tissue culture room. You should have respirators on hand where needed. And there should be project level risk assessment for cell sorting experiments. So in some ways, this part of flow cytometry is often best left to the experts. There are benchtop sorters, but do not neglect these risks or do so at your own risk. So there are two very basic types of cell sorters. And this is a historical sort of slide. The first is a jet and air sorter. These were developed in the late 70s. There are a couple of differences between a jet and air sorter and a cuvette-based sorter. A cuvette-based sorter uses a quartz cuvette and gives you a better signal resolution by passing your signal through the optically matched quartz cuvette and then generating the stream, whereas a jet and air sorter generates the stream and then actually reads out the fluorescence. That gives you a little bit more certainty in which drop is going to contain your cell of interest on a jet and air sorter, and a little less certainty on that on a cuvette-based sorter. So once you've gotten your fluorescent signal, you need to form drops. And there's a piezoelectric that chip that forms these drops at a resonant frequency. That resonant frequency is dependent on the nozzle size and the drop drive frequency. And then the amplitude is set to maintain a stable breakoff. That breakoff position, the last connected drop, needs to stay completely constant in order to actually accurately sort your cells. You'll see this stream camera on every cell sorter. The stream camera is viewed using a strobe and some image processing, which is why even though it's moving, it doesn't look like it's moving. If it does look like it's moving a lot, and you need to stop sorting. Critical for sort accuracy is to calculate the microsecond delay from the last sensing of the cell, so where the lasers intercept the stream, to where the last drop is connected. That value is called the drop delay. If your drop delay is incorrect or even a little bit incorrect, it can really reduce the purity and recovery of your resulting sample. And you can see this in a nice paper of which Rui Gardner is an author on a method called RMAX. Purity will stay pretty high across a reasonably large range of drop delay. However, recovery of your samples, how many sorted cells you get back out of the number you think you sorted, will drop off quite quickly. So your drop delay calculation and the accuracy of that calculation is pretty important in terms of getting back the cells you need out of the sorter. So next is droplet charging and deflection. Once we've identified a target cell, there's a charge that goes down the stream and breaks off into that first droplet. We can charge multiple droplets in order to increase the yield that we'll get off the sorter. Those charged droplets, once they have been charged, can be passed between charged plates. That deflects them left, right, and away from the uncharged droplets. Uncharged droplets will go to waste, as you can see here in the center, and you can get distinct side streams up to about 6 of them on modern instruments on either side. Those are just a different charge level that's put into the stream when you go ahead and charge the drop. So the sorted cells should form a visible side stream on your actual sort. This particular one is just a test stream, but you should see your cells heading into the tube when you're sorting. So the metrics associated with cell sorting, we should be able to put numbers on all of these things. So everybody will think of sort purity, right? Sort purity is very important. That's the percentage of target cells that are in your sort product. So you should be able to approximate, in many cases, 100% purity. If you are not at 100% purity or close, you should be looking back and saying, how can I improve this experiment? The point of going through a cell sorting experiment really is to get your cells very pure for the next step of your work. Efficiency is the next thing that we like to think about with cell sorting. And efficiency is of the how many target cells were you actually able to sort out of the number that were present in your tube? Or what is the percentage of the sort count minus the abort count over the target events? I'll go through a few of those terms later on. But your efficiency is going to be part of the readout that you can see while you're sorting. It'll be within the sort stats. And Where your efficiency should be will vary depending on the sample that you're running and the speed that you're running at. However, keeping that efficiency high is what's going to cause you to have good recovery. Recovery and yield is the next one. This is the percentage of sorted cells that are actually recovered post-sort. So if you took your sorted cells and you went and counted them, how many of those cells did you get back from the sorter? Lots of factors can actually cause problems with the yield and recovery count because there's things you need to do to concentrate your cells and to count your cells that can have a lot of error in them. So I wouldn't be surprised if there's some differences between what the sorter says it sorted and what you actually were able to count when you got back to your lab. However, I think that those should be a predictable and consistent number optimized to your method. The sort count is the other one. That's the absolute number of sorted cells, and that could be determined based on what the sorter gives you, or which is based on efficiency, or the yield or recovery, what you can actually see in your tube after you get it back. So now we're going to move to FAQ, and there's quite a lot of these because I've been doing this for a long time. So I get the same questions, and I've just put them into slides for all of you. Okay, so what are the buffers and media that we use in cell sorting? The first thing is sheath fluid. This is what we refer to all the time. Usually it's just buffered saline solution. On analyzers, you can use water, but on a cell sorter, because you need to charge your drops, you need to have conductivity. So usually it's just PBS or another type of saline. There's your sample buffer. What I would recommend to people is that they bring their samples in PBS with low protein. You can also bring them in media with low protein. You do need to make sure there's no clumps or particles of any kind. A lower protein than you use in complete media will help the cells, keep them from clumping and help them maintain viability, but it won't increase the viscosity, which can impact your sort. Your cell concentration will vary by application, but it usually varies between about 20 million and 0.5 million per mil. You should, if you're using a core, consult with them, and if you're not using a core, see what advice you can get from experts to help you optimize these parameters and make the most of small samples or rare cells or any more challenging sort. There's also the collection buffer. This is the buffer that you put into your collection tube, and it's intended to cushion the landing because the cells come off the sorter moving pretty fast, and we don't want them to jump into an empty swimming pool. We want to have something in that collection buffer that's going to help them keep them happy. So often it's complete media. Some people will use high serum media. I personally used to sort into PBS plus 50% serum. This is a good way to go. You're going to dilute that. The more serum you can have in there, in my experience with lymphocytes anyway, they recover very nicely that way. You also can sort directly into an RNA DNA isolation buffer or an RNA later kind of thing. This will stop the cells at a point where maybe your transcriptomics are pretty stable and may be a good way to go depending on what you're looking to do. We have a lot of choices on that. The only negative on sort of an RNA DNA method there is that you're not going to be able to assay the cells after sorting. So you're just going to have to take on faith that you got back what you thought you did. And then the volumes for what you should collect into, at least 10 microliters in a 384, 25 in a 96, 200 microliters in Eppendorf tubes, 1 mil in 5 mil tubes, and 3 mils in 15 mil tubes. Now, we go lower than this all the time. You know, we sort into 4 microliters in a 700 microliter Eppendorf tube, but you really need to be very careful with targeting if you do that and to make sure that your cells are landing in that collection buffer where they're going to be recoverable. So another great question, since everybody sees the drops, whether they know how the sorter works or not, the drops are up on the screen. Does every cell contain a drop? No, unfortunately, they don't. So there's some uncertainty in the cell position at the first drop breakoff that can influence the sort mode choice. For most sorting, about 1 in 4 drops can contain a cell, or they get too close. So if they get too close, they're not sorted, and that drops your efficiency, and you'll end up skipping those target cells. Those target cells that are skipped are called sort aborts, and they will be reported in any kind of sort report you can find. So the maximum event rate is going to depend on a lot of things. Basically, how many drops are being made, which is the nozzle size, the pressure into the system, the drop drive frequency, and the amplitude. So on a 70 micron nozzle, you can get a max speed between about 16,000 and 20,000 events per second, depending on your drop drive frequency and your pressure. At 100 microns, you can get between about 6,000 and 8,000 events per second. You go higher than that, you're really just dumping more cells into the waste, and it's not, you end up kind of tipping point of you're using up your sample, but you're not getting more cells back. Okay, so a lot of cell sorters today use the term flow rate, and I find flow rate a little bit of a confusing term because the cell sorter has a speed that you can call a flow rate, but it's not a volumetric rate. So the cell sorter doesn't run 1 microliter per second or anything like that. It's a pressure differential between the sheath pressure and the sample pressure. So the sample always has to be injected into the sheath at a slightly higher pressure in order to push the samples into single file line in order to do hydrodynamic focusing. So as you increase that pressure differential, your events per second go up. That's why people do call it a flow rate. And the diameter of the hydrodynamically focused core stream also goes up. So as you can see on the right, that means your cells, as the core stream goes up, do not line up as nicely. So you lose some resolution in your fluorescence signals when your cells are not as nicely, clearly in single file. This can result in coincidences, particularly at high speed, as cells have more space to wiggle around in the core stream. So in general, I think you want to set up your samples to run at the lowest possible flow rate that you can while keeping them in suspension and not aggregating. And that is going to be an experiment-specific thing that you need to think about when you're planning. So another thing is, how do I set the threshold for cell sorting? I think most people in this room, and hopefully most people who are watching online, will know something about what a threshold is for flow cytometry. It's basically the minimum intensity value that you can set to cause the cytometer to think that something that breaks the laser beam is an event, not a cell, an event. Every particle below the threshold is just invisible to the sorter. This doesn't matter as much in some ways on an analyzer, but on the sorter, anything it can't see will be randomly distributed to the sort collection. And your downstream application will really have a lot of influence on what you want to, where you want to set your threshold. So it's often set on forward scatter height. You can also use fluorescence. That's application dependent. I've got 2 examples of a very low threshold here, where you can say that all my happy cells are right here. They're the nice purple ones. And I've got some tissue debris and other stuff, cell debris down here. In this case, I've got a higher threshold. Now, why would we do that in 2 different sorts? One reason that we would do that is because in this particular case, we're isolating cells for something where we want to be sure we don't get any dead cells into it. So we might be doing RNA-seq on these. In this case, we're going to do a co-culture experiment. And whatever's dead in that 7-day co-culture is not going to make any difference. You're not counting it when you decide how many cells you're putting into the wells. So those dying cells are just not going to make a big difference. That's, again, just application dependent. So think about what your downstream application will be, and that will help you determine where you want to set your threshold. For tissue debris and like digests of a tumor sample or something like that, you get so much tissue debris, you really have to be careful that you don't lose your cells within by setting the threshold. Okay, so another thing, some people, when I learned to do analytical flow, people weren't really doing as much doublet discrimination in analysis. I think in most cases, certainly people who work with cores at this point do doublet discrimination. This is a method where we use the raw pulse shape data of the event in order to determine whether we're looking at a single cell, which would have a higher height and equal width, or I'm sorry, and a higher, would have the same height and a increased width or area compared to a doublet. And so the way that you set those gates is here you're looking at forward scatter height versus area after you've done your scatter gate, and you're setting a gate that is going to exclude these cells that have higher area than height, because those, because of the raw pulse shape, are going to be doublets. Now, doublets can be 2 cells stuck together, or if you're running fast, they can also be what we call coincidence, which is 2 cells that happen to be both counted as one event. For sorting, we usually use 2 sequential doublet discrimination gates. So you're also going to set gates a little bit differently in sorting versus analysis. In sorting, your gate logic is extremely important, and also the way you draw your gates is important. So for analysis gates, a lot of people use quadrants. It's not our favorite kind of gate in 2022, but a lot of people use quadrants, and this is a reasonable way to analyze this particular plot. If I were sorting these cells, I would want to be a lot more stringent in how I'm identifying these cell types. So for a sorting gate, if I use quadrants, I'm going to have a lot of cells that are on that boundary. We don't know what's going to happen with those. I'm going to have a lot of noise that's included. These are not extra big B cells here. This stuff here is not important to anything. We want to make sure that we're including exactly the cell types that we want. So we want to be pretty stringent and restrictive with the gates. Your gate logic is important because you can, more so than in an analysis method, run into a situation where you're trying to sort cells that are actually falling into more than one sort gate. So in this particular example of gate logic, this is from a BD FACSDIVA instrument, and you can see that the sort populations for this were the naive cells for both CD4 and CD8, and these are mutually exclusive. However, if I tried to sort CD8 naive and CD8, I'm going to get a lot of cells that are not sorted because they belong to both of those populations. So you need to think through the logic and be sure that your gating is mutually exclusive, that none of your populations fall into branches of the same gating tree. Okay. Generally, I would set the gates so they don't touch because this will increase aborts, and just make sure that you're very careful about the gate logic. Use double negative gates if you're moving to the next plot. So I'm going to give you a very simplified model of what a sort mode is. This probably was true years ago and is not exactly true now, but it's a good way to think about what a sort mode is. So a purity sort is the most common thing that people will think about. This used to be, and still is in some cases, a 2-drop envelope. What that means is that every time I find a target cell I want to sort, I'm going to sort 2 drops because I don't know if this cell, which is near the merge, is going to be in this drop or in this drop. So I'm going to sort both drops. I can also set up a 4-way or multi-way sort. This could be a 1.5 drop sort. Now this case, if I were looking at this cell which is centered in the drop, I might sort this one cell. However, if I was looking for this one, I would sort 2 cells because I had some uncertainty on whether this guy was going to break off down into this drop or come back up into this drop. Okay, so the sorter is not sorting half a drop. It's just making decisions about whether it's how many drops it's going to sort based on its certainty of where they're going to fall. There's also sorting mode that you'd use for plates or any kind of single cell sorting, which is often called single cell mode. This is a 1-drop sort envelope, and in many cases it uses a drop center or a cell centering portion to it, where it excludes cells like this one that are going to be not centered within the drops. So it's measuring where they are within tenths of the drops, and it's saying if it's in the first 2 tenths or the last 2 tenths, we're not going to sort that. You'll miss a lot of cells if you do a single cell sort, but you'll also be very confident that what you get is what you expected to get. The last sort mode, which is kind of in many ways underutilized, although it's not for every sort by any means, is a yield or enriched sort. This is a type of sort where you can take a rare population and run it through and just cause there to be no aborts. So every target cell that you want to sort, regardless of what's near it, what's in the same drop, what's in the next drop, will be sorted. So coincidence events don't matter in that case. You just need to set your gate so that you're able to isolate the cells that way. And one positive thing about that is that it can be used for quick debulking. You can run a lot faster if you're doing it on enriched sort. And Veena will tell you a little bit more about sort modes in a few minutes. So that's all I have on that. So how can you expect 100% purity or near 100% purity when you're sorting a very low frequency population? Well, there is a drop in, you can get down to maybe 5% of your initial frequency per population and still expect high purity. But as you get below that, your purity is going to drop from 99% down to 95, 93. As you get to 1%, you're still going to be down somewhere in the 60s, 70s. As you go lower than that, in one round of sorting, you are going to start to have less purity. We can only enrich with the sorter. We can't purify if there are very, very, very few cells. But you can go from 1 in a million to 1 in 10. Okay, so you may want to think about enrichment strategies before sorting. And I know Simona Cola was telling you about max enrichment, which she does for her hematopoietic stem cells. There's also kits now that you can use for dead cell removal. You can do enriched sorting, as I mentioned. You can also increase your initial frequency by doing what's called index sort gating, and I'll show you an example of that in a little bit. So a few other fake graphs here. These are relationships between nozzle size, pressure, efficiency, and events per second. And what I'm showing you here is that as you increase your nozzle size, your sheath pressure always comes down. You need to find a resonant node, and your sheath pressure is always going to drop as your nozzle size increases. Your drops are bigger, and your pressure will be lower. Your efficiency is also dependent on your events per second. So I told you before that if you run near the maximum events per second at a 70 micron is about 20,000 events per second, your efficiency will drop from about 100% in many cases down to somewhere in the 70s. So you need to think about where you want to be in terms of efficiency. If you have plenty of cells to spare and you can afford to throw some away, you may want to run faster. It's all a balancing act. If you choose a different sort mode, this slope here is going to change. So nozzle size is also related to the maximum events per second, as I said before. So at a 70 micron nozzle, we can get up to 20,000 events per second. If you get all the way out to a 130 micron nozzle, you're going to be down in the 1,000 events per second maximum. So a bigger nozzle is great, and it's maybe less stressful for your cells, but it's also much slower, so your cells stay out of their native state for a longer period of time. It's a balance how many cells are going to come out alive if you really increase the nozzle size, drop the pressure, drop the rate, spend more time on the sorter. Okay, so that's all my FAQs. I'm going through quickly here. Next thing I'm going to tell you about is how to prepare a good quality sample. And this is not exactly a how-to, but it's some considerations on how you might approach this. So a high quality single cell suspension is the single most important factor that most people can control in their sort results. Possible things can happen with the sorter, but things mostly happen with the samples. If you have a sorter that's run by experts, you're much more likely to run into trouble with problems with sample preparation than you are to run into problems with the instrumentation itself. Okay, so don't clog the nozzle. This is the key to any successful sort experiment. This is the same diagram I showed you before. And last time I showed you this, all the cells were nicely lined up. Once they don't line up and something plugs up that 70 micron or 100 micron orifice, a clump of cells, what you end up with is a little bit of stuff can still pass through. So you get a spray of aerosol that comes all out of the sorter. It gets into the collection tubes randomly. It contains cells. It contains cell fragments. It contains whatever else happen to be in there that you do not want to be breathing. So clogging the sorter can easily ruin your experiment. You do need to really think hard about biosafety. If you're trying to be safe, just don't clog the sorter. There's simple things you can do to limit the chance that this is going to be happening. You're also going to contaminate your sort product in many cases. Lots of modern sorters have ways to close off the collection tubes if you do encounter a clog, but that still is going to impact the purity of the sort population. You're still not going to be able to close that off quickly enough with automatic sensing that you're still going to get a 99% pure population in your sort product if you've had a clog. Clearing the clog in itself is not just a hassle, but also can change some of the QC parameter factors with the instrument. So sometimes it doesn't come back to exactly the same place that it was. And that means that some of your parameters can change intensities. You may need to move gates a little bit. Your break off may move a little bit. Clog prevention, again, just please make your samples good enough that they don't clog the instrument. Start with a good quality sample, a clean sorter, filter your samples through 40 micron mesh before you load them on the sorter, and certainly look at them under a light as well. Make sure you don't see anything floating in the tubes. If you can see it, do not put it on that sorter. Okay, so your quality of sort samples is extremely critical for hydrodynamic focusing. It's critical for forming good drops because all kinds of extra stuff will make your drop shape change. It will cause them to behave strangely. They don't deflect as well. Contaminants in the cell suspension can include aggregated cells, free DNA, tissue debris, red cells, lots of things that can get in there. Your cells within the stream need to be staying centered, and any kind of larger bolus of stuff is going to stop that process. And that's going to impact your fluorescence signals as well as your actual sort. So some tips on how to get a good single cell suspension: filter everything last thing before sorting. Filter through 40 micron mesh every single sample before you put it on the sorter. Watch the stream break off. If you see it move, the sorter might be monitoring for it to look for it move, but if you can see it move, it's moving. Your event rate should stay stable during sort. So if you're started at 5,000 cells per second and it's bouncing down and up and down and up to 1,000, 7,000, take that sample off, vortex it, refilter it, put it back on, see if that helps. Consider diluting that sample. Use a low concentration of protein. About 2% FBS is good for a lot of cell types. If you don't like serum, there's some other ways to go with that. Also use 2 millimolar EDTA. It doesn't seem to hurt any cells that I have located, and it does inhibit calcium-dependent aggregation. And then figure out what you can do up front to remove your dead cells and tissue debris. And some of this may just be washing your samples repeatedly. If you can see pieces of debris, you're not getting extra cells out of that. You're just making your sort worse. Okay, so for sorting, you also should always be doing viability staining. You want to make sure you exclude dead cells from your sorts. If you are including dead cells, then you're always going to end up with sort of a progression where cells are dying, your numbers are inaccurate, and your dead cells are making the live cells a little less happy. Dead cells have unexpected fluorescence. Once they're dying, they start to fluoresce in weird places for reasons that are not related to your staining panel. You can use a couple of different types of viability stains. These would include DAPI, amine reactive live dead cell stains. These amine reactive dyes need to be stained with your antibodies. Something like DAPI or Cytox or PI can be added at the sorter. If you don't forget that you need a single stain for your live dead control. It's a fluorochrome. It needs to have its own control. You will need dead cells in that control in order to do the compensation, and it can be a roadblock if you don't have any dead cells in your viability control. If you have very low viability or a lot of tissue debris, consider using calcium AM. Calcium AM is the only live dead marker that stains your live cells, not your dead cells. Always better to pick a positive signal than a negative signal, because most things that are events that are not cells are negative. Okay, so how do you adapt a staining method for cell sorting? You're going to want to stain, you're starting with a lot more cells in most cases. So you're going to want to stain in the smallest volume that you can get with your cells. So usually that works out to somewhere between 5 million for a big cell and 30 million per mil. Your antibody titers, if you get them from the companies, are usually weight or volume per 100 microliters of staining buffer, assuming 10 million per mil cells. So you're going to need to scale up the amount of antibody you use, but if you're using 1 million cells for analysis and you're going to use 10 million cells, you don't need to scale up by a factor of 10. You might want to scale up by a factor of 2 to get a decent titer. If you have an analysis method where you have very carefully titered your antibody, then you're going to want to scale up linearly. If you're using the minimal amount of antibody to get good resolution, then once you increase your cell number by a factor of 10, you're certainly going to need more antibody to maintain that good resolution. A lot of times people are starting for non-antibody methods with something that was developed and written, a protocol that was developed and written for analysis. And it's important to consider, again, that your cell numbers are probably higher for cell sorting, and you may need to think about things like time and volumes and the amount of dye available in order to get decent staining for this kind of approach. Okay, so we got through how to prepare a good quality sample, which is extremely important, and I hope everybody realizes the importance of this. This is the reason why experiments fail. Okay, so now we're going to talk about batch planning and applications of cell sorting. So I'm always going to tell everybody to plan ahead. Consult with the core if you're using a core. Look at the instrument that you're using if you're not using a core. Order your reagents. Make sure that the sorter you're going to use can see the fluorochromes that you've chosen. Write down a protocol. Outline the controls that you're going to need. Then run a pilot experiment, at least one. Try this before you run the real experiment. I have people who come into our lab all the time. They want to run something that's going to cost a lot of money, and they want to run it first time with all the mice they have. This is not a strategy for success. Try it first. Try it with something that doesn't matter. Identify the problems with the panels, the protocol, the antibody titers, and the controls. You're going to have enough trouble on the second experiment scaling up to a bigger experiment. So at least have run through with a successful result on the pilot experiment. In sorting, you often have to decide real quickly what you're going to sort. So in analysis, we can usually just record the data and then try to fix it up later. In a sort, you really need to kind of make a decision while your cells are still alive on what populations you're going to sort. So test your protocol before you start with critical samples. Often you can do pilots with things like PBMCs or normal mouse spleen, even though they don't exactly match what your experimental protocol is. They're normal cells that will stay with a lot of people's panels that are easy to get. Okay, so we can help you find the cores, experts, anybody can help you find a way to test protocols without pulling up the very important cancer sample that you can't possibly replace. What kinds of controls do I need for cell sorting? Well, so the answer for this one, unlike almost every other question I've answered, is almost exactly the same controls that you would use for an analytical flow cytometry experiment, and all of them. So you need your unstained cells, you need beads that aren't stained if you're using single color beads, you need your single color controls. I like comp beads. I know there's a lot of provisos with them, but they're reliably, consistently is a positive population. And if you can't get a positive population, you can't do the math to do either compensation or unmix. Do not forget to include your viability stains. These are dyes. They don't stain regular comp beads. They don't stain live cells in many cases, so you have to have some dead cells. You need gating controls. These are usually FMOs. Once you've run your FMOs, you'll know where to place your gates more confidently, and you'll be able to proceed and with confidence that you're identifying the cells of interest. You should also consider all the biological controls that are relevant to your experiments. So you might have parental cells or wild type cells or normal PBMC or similar tissue that you might want to look at. If people are looking at mouse tumors, I always suggest that they look at mouse spleen because often they're looking at immune cells, and immune cells can be found in spleen, and spleen is easy to get and much cleaner than any tumor. So another fun question, how many cells and how much time do you need? I used to work with somebody who his answer anytime anybody asked him what's the minimum number of cells you can sort is 2. Got to bring 2 or you can't sort them. Anyway, so how many cells, the way to calculate this is relatively intuitive, but how many cells are you going to need for your downstream application? Take that. Look at the initial frequency of your target cells. This should tell you how many total cells you need to bring to the sorter. Don't assume 100% yield, assume something like 70% yield. This just gives you an extra cushion to be able to say, I can still do the next step in my experiment. Once you know how many cells you need to bring to the sorter, you need to figure out what event rate is reasonable with your nozzle, your DDF, and your cells. This will help you estimate the time the sort will take and to optimize that cell per mil concentration to bring to this sorter. You can consider, we do this a lot in my lab, running in parallel on multiple sorters. Sabrina Bertolaccio told you yesterday about this, and she consistently will book 3 or 4 sorters for 3 or 4 hours all at the same time in order to get an experiment done. It's an efficient way to keep her cells alive and gives her good results and enough data and time that she's not spending 20 hours in the flow lab, and my staff is not quitting yet. Okay, so think about your specific downstream application. If you're putting your cells back into culture, which is like the old school, why would I sort cells? I'm going to sort them because they have GFP, right? If you're putting your cells back into culture, you can think about purity and recovery being possibly the most important things, purity probably being the most important. You'll need to find a compromise that's appropriate for your initial frequency. If you have very rare GFP positive cells that you're trying to sort, then you're not going to get to a 99.9% purity. So you should think about whether or not you can, what kind of compromise might be appropriate, whether you want to take those cells back and do another round of sorting after you've expanded them again. The cells per mil that you should bring for cell culture is going to depend on the nozzle size, which is somewhat dependent on the cell size for the cells you're going to sort. And you need to think hard in this case about sterility and carryover between samples. So make sure you clean the instrument with bleach and detergent, followed by water or PBS. It's always good to put it in a Quire on a tube of water. Make sure that you're not seeing events. you want to, if you're going to do single cell plate sorting, you have to have a pure population. You typically want 1 cell per well, so use your single cell mode. Bring your cells at a lower cell density, a lower cells per mil. And that would be so that you miss fewer cells because you're going to run slower. And again, consider index sorting if your cells are rare, because that's going to increase your initial frequency. Okay, more modern things you can do, or more, I guess, fashionable things you can do with sorting. Plan for your specific assay. If you're doing functional assays, lots of cells do well after sorting for co-cultures, for all kinds of different functional assays. You may need to work it out for your particular cell type and your assay, but I would tell you that if anybody tells you that cells cannot be run in functional assays after sorting, I can prove them wrong. So consider sorting directly into the culture plates when it's appropriate, although do realize you'll be adding some PBS when you sort into culture plates. If you're sorting for animal models, either a cell line derived xenograft or a patient derived xenograft, you're going to need an accurate sort count, and you need that to be aseptic so that you're compliant with all the IACUC things. And if you're sorting donor cells from PDX or CDX animals that have already been produced, then plan your panel carefully and consider what you're going to do with those cells after the fact. It can be difficult to identify PDX and CDX cells, and lots of times in a patient-derived xenograft, those cells can be quite rare. So it's good to have a decent idea of what to expect before you get to the sorter. If you're purifying cells before omics, you're going to need very high purity and low collection volume. Your accurate counts are probably very important. So you might want to consider single cell mode for that, and that would include any of the omics applications that we discussed the other day, and I'm not going to cover that as well as they did. Okay, so applications for cell sorting. Now we mostly know how this works. So we can usually sort, we often sort into tubes. These will be between 1 and 6 ways. I call this bulk sorting. So we're just not separating them as single cells. We're separating a bunch of types of cells into a tube. So for best results, bring your cells in a cell per mil, appropriate choice of nozzle. So I would tell people if they're sorting at 70 microns, they want to bring those cells to us between 20 and 30 million per mil. If you're sorting at 100 micron, you probably want to bring your cells at about 10 million per mil. It's always easier if you're in the flow lab to dilute your cells than it is to concentrate them. So bring some buffer to dilute them and make them a little bit more concentrated than you think you need them. Your choice of sort mode is going to depend on what you're doing with them downstream. How important is purity, yield, and single cell. And you can isolate up to 6 populations at the same time for heterogeneous cell suspension. And we can also isolate as many as tens of millions of cells in a single sort. So we can sort as few as 1 cell. Sorry, then we're coming back to that silly joke. And we can use about up to 40 floors to define complex phenotypes. The spectral sorting is coming online, and it's going to bring us up to that 40-ish space in terms of complexity. And we will keep you posted on that. So cell sorting is suitable for almost every kind of downstream application. So plate and slide sorting, we can do single cells or fixed number of cells. We typically will use a single cell sort mode for this. It just tightens up where the cells are going to land, as well as a few other things. It's an alternative to people do limiting dilution assays for CRISPR libraries these days, and clone isolations, and that leaves you culturing a ton of plates, and you may not want to do that. So putting them on the sorter and getting your cells of interest plated for you is a better way to get a single cell per well. For any kind of plate sorting, you're going to want your cells relatively at a lower cellularity. And I keep saying index sorting here, sorry. I'm going to go to that in the next slide. So you can use that for small samples like a patient biopsy. Some sorters can be set up to do simultaneous sorting of different populations into different rows on a plate. This is not especially common, but there are sorters that can do it. Check first to see if this is something that you need to do. Okay, so we can do a lot with complex phenotypes. This is actually data from Sabrina Bertolaccio's lab. And what we're doing here is we're looking at her myeloid cells, including a lineage cocktail, and we have 4 sort populations. So her non-conventional, intermediate, and conventional monocytes that come from the Lin negative gate here. So those are here in this plot. The Lin positive gate here goes to the MDSC. Both of those are preceded by CCS CD66B negatives. So what we want to be sure here is that we have good gate logic. And the way I know we have good gate logic is that all of these gates are mutually exclusive. So these do not overlap, these do not overlap. And it's helpful if your lineage positive and negative are both drawn on the same plot, because you know they do not include the same cells in that case. So we want to make sure we're using mutually exclusive cells. In a lot of cases, you might want to use a gate that is a not gate in this as well. So she's got, starting with about 0.5% to 1% initial frequency, she'll get high enough purity to get downstream RNA-seq. And she uses a lot of controls, including biological controls, to determine the gate positions here. So, Veena's going to give a talk in a minute on rare cell sorting. So I was just going to say we can do rare cell sorting, and stay tuned for Veena's awesome talk. Index sorting, which I mentioned, this is kind of a cool thing. You can take rare cells that you would typically have trouble sorting, and you can set gates in such a way that you are using Boolean operators or a sort of hacked up Boolean operator. This is going to be an AND gate here that's shaped like an L. And you're going to say, all of these are my target population. Put them into a plate, it's single cell per well. And we're going to have an index where we can say, well A1 got a green one, and well A2 got a purple one. This is a good way to increase your initial frequency, which is going to increase your eventual purity of your sort product, as well as your recovery of your sort product. This is underutilized and is a thing that should be used in many applications. That retrospective knowledge of which cell went into which plate is also applicable to cells that are differentiating on a continuum, that are not exactly defined to a particular cell type. Minimizes everything going to waste if you are able to take this strategy. And we're hoping that soon we're going to be moving this into a multi-omics approach, similar to what Trisha told us about yesterday. There are some roadblocks in multi-omics. I think we're taking a different direction than she had. We're going to be getting the Rhapsody system in the lab, and we'll be using that for multi-omics. Okay, and that's all I had to say, except to say thank you to the great people who work in my lab, and the great people who used to work in my lab, and all the amazing flow Texans who are here today, as well as a few other people I worked with closely for cell sorting and learned a lot from. So thank you very much.
Okay, that was great. Just so you know, Karen, you had a lot of very positive feedback in the YouTube chat. And you've been volunteered to write a paper. So we're going to save, if there's any questions, you can ask them at the end, because we're going to do a roundtable. And same thing, if there's any questions that are coming from YouTube, go ahead and put them in the chat, and we'll get to them. Is there? Okay. All right. Our next speaker is Veena Papanna. Veena is a flow cytometry coordinator at the University of Texas MD Anderson Cancer Center Advanced Cytometry and Sorting Facility. We've seen a few people from there. She received her master's degree in microbiology from the University of Bangalore, India in 2002. Veena has more than 10 years of biomedical and immunology research experience, including over 5 years of experience working in flow cytometry course, probably more at this point. She received her cytometry certification from ISAC in March 2015, and she's attended many hours of cytometry training from BD, ISAC, FlowCyte, like Amnis, including a lot of software training too. She has been associated with the ACSF at MD Anderson since 2011, and today we're going to hear her talk about rare cell sorting, as Karen just mentioned. So please help me welcome Veena.
Thank you, Sarah, for the introduction. Good afternoon, everyone. Today, I'll be talking about rare cell isolation strategies. This is an ongoing project, and I have done just initial experiment, just a couple of experiments, and the results was pretty interesting, so we thought of sharing. So here, the first slide I'm showing here is the rare cells, just some examples of rare cells is cancer stem cells, circulatory tumor cells, or circulatory endothelial cells, or hematopoietic stem cells, and etc. So the name suggests the rare cell is a rare population which is low in percentage, and despite low abundance, a rare cell population play an important role in determining the pathogenesis of cancer and mediating immune responses and angiogenesis in cancer and other diseases. So objective of our project is to isolate rare cells and to know the purity of the sorted cells and understand the sorting efficiency and develop a rare cell sorting procedure or a protocol to educate the researcher on the ability of our sorters. So methods, I have not given all the details of the procedure and sample preparation here, but the most important part is the isolation of PBMC. We did isolate the PBMCs from whole blood using Ficol and density gradient centrifugation, and APC beads, APC fluorescent beads from BD calibrite kit was added into the PBMC to mimic a rare cell. So we are not sorting a real rare cell here. So we are just mimicking the rare cells, and APC fluorescent beads will act as a rare cell here. So APC fluorescent beads are added into the PBMCs in a serial dilution method to create the sample, that concentration that we wanted to create. So here we have the serial dilution, how we did the serial dilution here. We did isolate the PBMCs, and final concentration was 20 million per ml in PBS. And we added in the first tube, you see, we added 3 ml of the that concentration, which makes it 60 million. And then from 60 million, we added APC beads, a drop of APC beads, and mixed well. And we did the analysis on flow cytometry instrument to get an absolute count, just to see, to adjust the concentration to 1%. That you can see this plot here. We did adjust the concentration to the 1.07%. It's not exactly 1%, but yeah, we were pretty close. And from here, this is basically our dilution 1 in 100. So we did the serial dilution in the 4 other tubes. So that would be 1 in 1,000, one bead in 1,000, and 1 APC bead in 10,000, 100,000, and 1 in 1 million. So theoretically, the APC beads that we had, according to the 60 million PBMCs we have added, the theoretical the beads that would have in that tube is 600,000 beads and 60,000 beads and 6,660 beads. So 60 million beads will have only 60 beads of APC in the last tube. Sample number 5. So sorting detail, this is our first experiment. The sorter used is BD influx, which you can see here. 100 micron nozzle were used and 27 psi pressure. And sort mode is sort. I would usually say sort 1 and sort 2 here. Sort 1, when I say sort 1, it is first sample is sorted first time on enrich mode. And then whatever the sort was collected, the target sorting is APC beads will be sorting. So APC beads were collected, and that sorted sample is resorted. That is sort 2. So usually I would just say sort 1, sort 2, but it's not that I'm sorting 2 different sample here. I'm sorting the same sample, and I collect the sample, and that sort the same collected sample on a different sort mode, that is pure mode. The first time I sort on enrich mode, and the second sort I sort on pure mode. So the event rate for the sort 1 was very quick. That is 35,000 events per second, and sort 2 is 250 events per second was run. And these are the samples that already mentioned about. So these are the 2 slides I just want to skim through because Karan has already covered this one. But I just want to say one thing here is purity is more important than recovery. whenever there is a non-target cell closer or in the drop, it will be aborted and it is not sorted. And only cells which is our target cells or in the middle of the drop was sorted. And enriched sort recovery is more important here than the purity. So in spite of having non-target beads or cells in the sorting the drop which is getting sorted, it still will get sorted in spite of having a non-target beads or cells in there. So basically the greater efficiency or recovery we do keep the sort efficiency on enriched mode. So these are the results for the first experiment. Initially, I did say that our goal was to see the efficiency of the sort and also the purity of the sort. This is like efficiency. This is the sort count for every sort. So you see the green, the column here, which is sort 1. So number of beads collected after this sort 1. And the second one is a bead collected after the resort of this samples. So here, the first, the range was from 60 beads to 50,000 beads. So I wanted to show everything in one plot, but I could not fit that. So I did convert these numbers to percentage compared to the target beads. So here you see the first bar here. You see these are the 5 samples with a different concentration of rare cells, rare beads here. So here you see the green bar here. The first 2 we did, because of the time, we didn't want to sort these 2 samples like completely, that is all 60 million cells. We just sorted and set the sorting count to 50,000 on this one and 10,000 on the second one. And both are 100% recovery. And the last 3 samples, we did sort a complete sample, 60 million cells, so that because it was beads were kind of low, so that's why we wanted to completely sort the sample. So here you can see that all the 3, like one gave 115% recovery, 77% recovery, and 153% recovery. So here kind of it gave us more than what was added, theoretical like numbers, like beads added. It was like not surprising to us because it is on healed mode. So first thought was in healed mode. So we were like some non-target events or beads would have entered, the PBMCs would have entered. So we were like fine with that. But when we sorted that sample again, second time with pure mode, one drop pure mode, then we see that the purity is 94%, efficiency. This is efficiency was 94%. And this was efficiency was 34% because instrument got caught. Unfortunately, instrument clogged, and then we did lose the recovery there. So, but we did get 92% here and 75%, 105% of recovery. So the most surprising part was 1 in million. When you see here, you see that 60 beads, theoretically 60 beads should have been there, and we did recover 63 beads there. So it was interesting to see these results, but we wanted to see the purity also. So next goal was to see the purity of the sort, and here you see that first row here, you see these are the sort 1 purity, and the second row here you see the sort 2 purity. So here you see the first purity is not that good. It's about like 47, 30%. And then the last one was 16.4%. So this is yield mode. So we did see some PBMCs has entered and the purity is not great. But the second sort, once we sorted this sample, we did the purity jumped to 99%, 95%. And the sample with 1 in 1 million was 46.2% the purity. So here I'm just showing the same results previous slide, but it is just in numbers and bar graphs. So here the purity of only thing is all 4 sample purity was 90%, above 90%, And here, only the last sample, which is 1 in million, or 1 in 1 million, kind of gave a slightly lower percentage of purity. But it did increase from this to 16 to 46, a 3 times increase, but it's not as much as this 4. So this results kind of, it was interesting for us to quickly set up a second experiment to see if this is, we could reproduce this, or like, is this one time, or like we wanted to see just setting up a next experiment, one more experiment before getting too excited about these results. So here, the second experiment, the dilution was similar, but only thing is we did skip some initial dilutions and wanted to add one more, because it did sort all 60 beads from 60 million of 1 in million the samples. So we wanted to see whether the instrument can sort 1 in 10 million. So we did started with 0.1%, which is 1 in 1,000. And then we did skip 1 in 100 and 1 in 10,000. And we kept 1 in 100,000, 1 in million, and 1 in 10 million. So same way, the concept here, we started with 100 million cells, and we did adjust based on by running in flow cytometry analyzers, and 1.1% is adjusted there. And from there, after we adjusted the first tube, we did the dilutions, and we did get the to achieve the concentration of rare cell that we want. So sort detail is pretty much the same. Just one thing is that sort mode is kind of different. Here we wanted to sort 3 times this time. So sort 1, enrich mode, and sort 2, again enrich mode. Take the sort 1 sorted sample and rerun that, resort that, and with an enrich mode again. And the third time we sorted the second SAM collected beads, resorted that, and on a purity mode, 1 drop pure mode. So the event rate is similar, high, and then it to 2,500 per 2,500 events per second, and then 250. And these are the samples that we used for this time. So the results are like this. So same way, I did convert this to percentage, and you see only the first sample was sorted on only 18,000, not the whole sample was sorted. There's 3 of the samples were sorted complete. And then here you see 100%. The trend was similar. The first sort, that is enriched sort, had a kind of a more, the efficiency was kind of more than what was expected. And then the second time sort, even though it was enriched, because it was sorting, resorting for the second time, we did get 90%, 96%, 87, and 110% here. And the third time sort of this sample again gave us 71%, 96, and 80, and 100% recovery. So here again, we were kind of surprised. The 10 beads we added in that 100 million, we did get sorted all 10 cells, 10 beads there. So this is kind of exciting for us to challenge the sorter and give more, like maybe 1 in 50 million or something like that next time. So this is the sort purity. This is kind of crowded. So I would just jump to this slide so you can see the bars here. This is the purity, sort purity. The first sort with enrich mode was kind of like 30s and like 58. And then it did this to jumped high to 90%, above 90%. And the purity sort was also 90% on that. And this was, I don't know if you have, I did mention that the previous experiment 1 was 46.2%. And it was such nice reproduction. It is like 46.1%, the same 1 in million sample. And this 1 in 10 million kind of, first one was 5% purity, and then gave 10, and then 13. From 5 to 13, again, it did jump 3 times, but it is not as much because it is only 10 beads. Even if there is, because I was using APC beads, which is small in size, the instrument threshold was not set on high. It was kind of low. Maybe there is some even instrument noise, or anything could just get the purity to that low. So, but still, the good thing was that we got the recovery and the purity pretty good compared to the rare cells. So the future plan is, as I told that we did use a BD influx. So in future, we want to use BD ARIA or Cytec Aurora just to see if the instrument makes any difference, and also use 70 micron nozzle with a higher pressure. Would this change any purity or recovery? And third thing is we want to, here we did use APC beads. And next time we want to use live cells, that is, it could be tumor or T cells or anything, because the viability is the one thing that we really want to test here, because 3 times the sample is going through the instrument for sorting. We don't know if the cells would die. So the health of the cells, we really want to see that. That would be our next experiment to check the viability. We want to spike instead of APC beads, we want to spike live cells. So as I already told that we want to challenge our sorters and to see if this, if we can set up to 1 in 50 billion or 1 in 30 million or 1 in 100 million. We'll see how our experiment would go. Finally, we would want to, at one point, we would really want to sort the real rare cell to see if we, on basis of these results, we can decide and develop a protocol to see how we could strategy how we could sort the real rare cell samples. The conclusion: both experiment, that is experiment 1 and 2, both shows similar results of the trend was similar. The purity increases from enriched to pure sort mode, and the purity of the sample were excellent based on the rare cell concentration, and efficiency of the sort was surprisingly very good. And this contemporary sorters are effective in sorting rare cell. And then promising results of these 2 initial test results encourages us to set up more experiment in future, and mainly to check the viability of the cell. That's it. And I would like to thank all the members of the FlowTex for giving me an opportunity to talk here, and our core facility members, and especially Clise-Dwyer. So as Sarah said, she has also influenced my scientific career, and she's basically gives all this idea and proposals for their experiments, and I'm just executing this experiments. And Karen Ramirez is our manager, and I really want to thank her because she is every step of this project, she does help us. And then Catherine Cox, like she and we both are actually, Catherine and me are involved in this project. She has shared all, we both share the project and work, and she has done as much as work as I have. And Dwyer and Will, David Dwyer and Will and Matthew, they are my colleagues. Without them, this would not be possible because, I work in core facility and we have a set schedule and a lot of customers. So they let me work on this project, and they do take care of customers and they work, and I really appreciate them for that. And finally, I want to thank Lisa St. John, who does order some samples for us for the project. There's some citation and reference I referred, and thank you all for listening.
There's no program. All right. So now we have our next speaker coming in remotely, and that is Peter Lopez. He's an associate professor of pathology at NYU Grossman School of Medicine. He started his career in flow cytometry in 1977. Pretty much, I think I've seen Star Wars that year at the age of 4. Oh, I'm sorry. Did I say that out loud? Working at the University of Rochester with Leon Wheelless, evaluating slit scan flow cytometry as a tool for pre-screening gynecological samples. He later directed a core laboratory at Fox Chase Cancer Center, Dana-Farber Cancer Institute, Aaron Diamond AIDS Research Center, and currently serves as the director of cytometry and self-sorting laboratory at NYU. He was influential in testing and designing the MoFlow, the MoFlow XDP, the ZE5, and the Bigfoot cytometers. His current work focuses on what he calls sorter-induced cellular stress, the effect of the cell sorting process on sorted cells. And his talk is the effect of droplet cell sorting on sorted cells.
Okay, thanks a lot for the intro, Joe. And for the little nudge there with the Star Wars, I get it. And greetings from New York, well, from upstate New York. I'm not in the lab today. I'm 60 miles north where I live. People in New York City think I live in Canada because I'm 60 miles away north of New York City. But regardless, it's a gray day here too, as many of you probably expect is typical of New York City. So I wanted to thank the FlowTex group for inviting me. I wish I could be with you folks. I enjoy your meeting. I've been there and really thankful for the invitation. So also, Karen gave a really good introduction to flow cytometry, and I think a number of people are asking on the YouTube chat to have that put up as a prerequisite for anybody walking into a flow cytometry lab. So I hope the FlowTex people can put that up because it really was great, and it actually helps me out because it fills in a lot of the gaps in my presentation. So in my talk, I'm going to be talking specifically about droplet cell sorting. Later in the talk, I'll talk about other types of cell purification processes. But the focus for my talk will be, does the droplet cell sorter do anything to the cells? I'm going to start the talk with a little bit of the history of droplet cell sorting. And I'm going to talk about this thing that I call SIX, sorter-induced cellular stress, and some specific observations we've made in our lab to what we call the metabolic SIX phenotype. And then, like I said, the last part of the talk, I'll be talking about things you could do potentially on droplet cell sorter to minimize this sorter-induced cellular stress. And I'll also briefly talk about some other technologies that can be useful. So this is the history part. So the first droplet cell sorter was not a fax. Hopefully folks recognize that Mac Fullweiler was the person who invented the cell sorter, and this was based on Dick Sweet's inkjet printing technology. And hopefully a lot of folks who are using Becton Dickinson cell sorters understand that the sweet spot is actually an homage to Dick Sweet. It's his last name, and it's been immortalized in the Becton Dickinson cell sorter setup. When you look at the sweet spot, you're looking really. Just remember, Dick Sweet came up with that. And that sorter didn't have fluorescence, so the F in fax was not there. That sorter was based on Coulter volume, not fluorescence. And I'll just put in my usual plug on fax. Remember that is the fluorescence activated cell sorter and shouldn't be used to describe the technology, although that's frequently done. So I don't want to say don't use fax because we all want to use fax, but just be careful when you're using it in especially in publications. So then we moved to the fluorescence activated cell sorter, which came after Mac Fullweiler's sorter. This was invented along with a lot of input from Becton Dickinson by Len Herzenberg, who's also at Stanford. One thing to point out here, these are some of the original drawings. You may notice both in the color drawing and in the black and white drawing that there are three droplets being sorted, and you might be inclined to think that indicates three individual cells of interest in three drops. It turns out that back in those early days, this wasn't the case. Because of the way that we did things back in the old days, again, I'm telling you a bit about the history, primarily because I kind of was there. We used to sort a drop and then flanking drops that were empty. This was because of the technique that we used for establishing the droplet delay, which was basically a measurement, a manual measurement using a micrometer and a microscope. And also because it guaranteed that we were going to get good purity, basically because things were, this was kind of the Wild West back in the early days. Things weren't quite as locked down as they are today. The electronics, it was cutting edge as it is now, but we were right on the edge of what the electronics could do and everything else. So this gave a little bit of a leeway. Karen talked about a 2-droplet sort where you weren't exactly sure where that cell was in that envelope. Well, it was not uncommon to sort multiple droplets just to be sure you got the cell of interest. So I use this term slow flow, and it'll be obvious in a minute why. But this was the way we sorted from the time of the initial fax commercial instrument release up until the early 1990s. And I just put these numbers here because today these are kind of I mean we can easily sort faster than 5,000 cells per second, but until the early 90s, that was breakneck speed. That was people would get very nervous about sorting that quickly. The pressures of the total system were usually about 20 psi or less. Today's systems can go 60 psi, 70 psi, even 100 psi. And as I mentioned, typically we'd be sorting a droplet where we think the cell is, and then 1 to 3 empty droplets on both sides. So you would routinely see people sorting 7 drops just to get the middle drop to have the cells. That would be if drop delay was if the droplets were wandering around and you couldn't get it under control, you would sort 7 drops. But more often than not, you would be sorting 3 just to be sure you got the cell of interest. And another thing, since I'm going to be talking about the effect of the cell sorter on cells, it was not uncommon to use almost half a watt of laser power. And that's what we used to use on our FAX2 system. Today we've dialed back on some of these laser powers, but one of the questions that we're going to talk about is, does the laser have any impact on the cells, on the cell after it's been sorted? So to give a quick illustration of this, this is typical. I mean, the basic cell sorting strategy, the technology hasn't changed all that dramatically from what Mac Fulwiler put together in the late 60s. So this drawing here, I'm going to use again later in the presentation to show the more current setup. But here we go, 12 psi with sheath pressure and just a little bit over 12 psi sample pressure. I think that's an important thing that everyone should keep in mind. Karen talked about this a little bit in her presentation, but overall, the system pressures and the sample pressure are not that much different. So sometimes we have people coming into the lab who say, oh, you're sorting at 5,000 cells per second. It's too fast and you're going to be destroying the cells. We should dial back and sort slower. Well, you're dialing back from 12.2 psi to 12.1 psi to affect that change. That's not a dramatic change. And that's probably not going to change anything related to the cells coming out on the other end. So just keep that in mind, this notion of just, if you go X number of cells per second, it's too fast and half of that is better. The overall pressure of the system hasn't changed. Now, I'm talking about an old system here, 12 psi, but this applies to even the current systems where we're using higher pressures. So there's a lot of things going on with these cells. And, I've been doing this, as Joel pointed out, since, before Star Wars came out. And it still amazes me, that this whole thing even works. That we get cells out of these systems that we can do things with. But, we've been doing it forever. So we have to, so it's just comforting to know that all of these things can be pulled together, all these different kind of technologies and forces, to get a great result that's really beneficial for scientific research. So there's all this stuff that goes on. You have to process the samples before you put them in the machine. You're doing all sorts of stuff to the cells just to get. You might have to be teasing them out of primary tissue. You have to stain them. You have to spin them. They're going through temperature changes. They're going in and out of the incubator. All this stuff already is a lot to ask for help. But then you're going to stick it into a cell sorter, and now the cell sorter is going to pressurize it. You're going to push it through a nozzle. You're going to decompress the cells. You're going to hit it with a lot of laser light. You're going to charge the droplets and so forth. So again, it's kind of amazing that this whole thing works in the first place. But the point here is that there are so many places where the cell can be negatively impacted by the process. So, even back in the early days, it was recognized that, and this is a slide from Joe Trotter, who has been really very, one of the leaders in the whole cell sorting technology, where it was known even in the early days that you can't put, if we remember, I think it's Shapiro's first law, or I think it's the first law, that you can't push a 51 micron particle through a 50 micron orifice. Well, the orifice size is important. And if you pick the wrong orifice size, it doesn't matter if you're doing, cell sorting back in 1980 or doing cell sorting today, you can just, destroy the cell because it's simply, you can't squeeze it through without causing some effect from shear force. So this was well known. But then things changed, and all of a sudden high-speed cell sorting became the norm. And this was introduced in 1994. I like to tell this story because I was part of this whole change into, at least I was there. I worked at Cytometry starting in 1996. And just really quickly, because I think it's nice to know some of this history, is that the MoFlow was not actually created at Cytometry. It was developed at Lawrence Livermore Laboratory as part of the beginnings of the Human Genome Project. And it was built with specifically to sort chromosomes. And what Geir Vandeneng was very influential in the design of that machine, as well as other cell sorters afterwards, like things like the Jazz and the Influx. He had a lot to do with. But this was a big deal that we could go faster because what Cytometry did was they basically licensed the technology from Lawrence Livermore Lab, and then they just refined it, embellished upon it to make something that could sort cells. And this was what was called the MoFlow, which was released in 1994. Now, once this system was released, questions started arising about, does the sorter impact cells? The question that came up, all of a sudden people were saying, well, cells can't withstand these conditions. Well, and I was kind of on the front end of this because I was out there talking about this, and somebody would always raise their hand in the audience and say, oh, the cells can't withstand it. I always wondered, why can they withstand one setting and not the other? And how do you know that that's the case? It was just kind of a gut feeling that people had. Oh, this is too fast. The cells are going to be destroyed. And that's because the cells, to do high-speed cell sorting, you have to make more droplets. You have to use higher pressures. And also you have to concentrate the samples. Again, Karen pointed that out about the speed, the concentrations that you might want in order to be able to attain and maintain these high throughput cell sorting. And there were some questions about that too. Could you concentrate cells? Would this concentrated sample behave well if it was being sorted for an hour? So this question came up, and one thing I just wanted to mention is this. I think there's this notion of metrics of cell sorting, and I kind of like to use the word jargon only because we have to be careful about how we we talk about these things. And I'm going to be honest that this slide here, I'm not going to say is the definitive way of defining these items. I can say that back when I worked with Cytometry, we tried to lock down these definitions. And in fact, there's a presentation at one of the, I don't know if it was called Cyto or if it was still called the ISAC Congress that I made. And we talked about something, really taking these terms and providing real definitions. And it wasn't met with a lot of favorable views from, I think, primarily from more so from the vendors, because we were at Cytometry, we were really knocking it out of the park at the time, and I think a lot of the vendors were a little bit nervous that our definitions would make their systems look poor. I don't know the rationale, but anyway, the thing I think that's important here is that, and the thing I'm talking about in this presentation is is the material usable after the sort? Will the cells perform as expected? And did the sorter just kill the cells? Now, there's a lot of things that we can do with cells afterwards. And, yesterday we heard a lot about single cell applications and genomics applications. So, you have to keep in mind what kind of performance are you looking for from these cells? For instance, in a genomic setting, cell sorting wouldn't be so great if it was ripping apart nucleic acids. Well, we know that that's not the case. So, it's important in a cell sorting experiment to keep in mind and ask, what are these cells going to be used for downstream? Even in a single cell genomic situation, we don't all have quite the nice setup that Tricia has at the Broad, where everything's happening within the same lab, and in some cases, people are sorting for 10x genomics, and then they have to bring the cells, maybe to another building. In our case, we're lucky it's in the same building, but in some cases, maybe not. And are the cells going to make it to the lab, to the next step without degrading significantly? And that may not be outright killing the cells by the sorter, but there may be, they may have a short lifespan afterwards. Just to point out here that, if we go back to the beginning of the high-speed sorting kind of revolution, not many people were publishing because the machines weren't out there. The turbo sort was an option for the BD Pax Vantage at the time, and the MoFlow was an actual system built from the ground up to go fast. So when people were asking questions about the viability of the cells and so forth, we didn't have too much to refer back to. There were some publications, and that was promising because we knew that people were using the cells afterwards and they would work. And this was before a lot of the single cell work was being done. So typically they were being used for some kind of a viable post-sort assay. Now, again, same picture that I showed earlier, talking now about today's system. I'm using the MoFlow with 60 PSI. So everything gets pumped up. The speed of the sheath and sample, and remember the sheath and sample are all moving at exactly the same speed, is upwards of 25 meters a second. So, we'll talk a little bit more about what that means, but that droplet with that cell is going to impact a collection vessel of some sort. So, is that in itself a big deal for the cell? And then the droplets, 100,000 droplets per second, means that whole environment that the cells are in is vibrating at 100,000 hertz. And, I think we all know that if you have a tube of cells and you stick it in a sonic bath, like one of these jewelry cleaners, they're not going to last very long. So, all of these things now are more than they were in the past with the slow flow, and it made people work. So I was on kind of the front end of this because I was working at Cytometry, and I was employee number 34 or 35, a small group, and they didn't really have anybody who was a flow cytometrist until I came on board. So I had to go out and try to convince people that, without a lot of people having published on this, that these things worked. And I had a little tiny lab. Actually, I had a room that I had to turn into a lab, but that doesn't matter. Regardless, the trick was, let me just show people what they want to see, that the thing isn't killing cells. So I got some jerkbait cells, and I tried some different concentrations because 10 to the 8 cells per ml wasn't widely a concentration that people would be bringing for cell sorting. But I figured, let me just put them all together and let's see what happens. And this is the type of numbers that I used to show back in the late 90s when I gave talks trying to convince people that this instrument actually worked. But it's only jerkets, right? And I didn't have a lot of options and so forth. But at least, some people saw this and said, well, yeah, it's not destroying the cells. When we talk about this high speed type of cell sorting, this is another slide that I took from the presentation that I made a number of years ago. But this was a slide that Matthias Scheinman contributed to the talk, where he's pointing out that 60 psi, which is the pressure that the cells are being put under, is the equivalent of what you would experience if you were 40 meters underwater. And that's, I don't go that deep, but folks who dive understand the consequences there. 90 kilometers per hour is about the speed of, it's not a race car speed. It's about the speed of a that you drive if you're obeying the speed limit. And I'm not going to say if I do or not, but if you're going 55 miles an hour or 60 miles an hour, that's about how fast the cells are moving through a 70 micron nozzle. And then there's also all of this laser that's impacting the cells as they go through there. But the one thing to remember there is the time period where the cell is being illuminated by the laser is very brief. One thing that we didn't talk about during this talk in 2018 was the droplet formation. And I put this up because this is something that we're feeling lately is something that may be a major contributor to the process of 6, which I'm going to describe. And there's a lot of amperage that's put onto the droplets to get those droplets charged so that they can be deflected. I don't know if you've had the experience of touching the charge plates, which it happens once in a while, and you get a brief shock. I'm not recommending that you try this, but if you've done this by accident, that's kind of startling when you have that shock. Being on the instrument design side, I've actually accidentally touched the charging side of the system, the side, not the plate side, the charge that's going into the stream to make the droplets. And I had gloves on both hands, and I touched the item where the charge pulse is applied, and I had my hand on the MoFlow bench, which is metal. And I'll tell you, that shock pretty much sent me home for the day. And it burned through the gloves on both sides. Just to give you an idea, that's kind of a personal reference, but there's a lot more. Even though the voltage is lower, the amperage is higher than what you experience on the plates. So this all leads up to something that we talked about during that 2018 presentation, where we came up with this, I say catchy acronym. I'll let you all in on a secret. We came up with this word 6 about 2 hours before we made the presentation. We weren't talking about it. It was in our presentation. We made a quick slide and added it to the presentation that we made at Cyto. It was my idea. Basically, I said, look, let's just come up with a catchy name for this. So like it or not, this is what we came up with. And 6 can, so it's sort of induced cellular stress. And it can present itself as a spectrum of symptoms, or it could be outright cell death. So it's a whole range of things that can happen. And there have been reports, some published, some made as presentations, about things that were happening to cells after cell sorting. Dendritic cells not presenting antigen effectively, T cell subsets not growing very well, but eventually growing, redox changes. There's a thing called a tetraploid blastocyst complementation assay, and induced pluripotent stem cells didn't seem to work for that. So there's any number of these very specific outcomes from cells that are being put into some other assay afterwards and expected to perform. And the point that I'd like to make is that we need to be aware that there's some potential complication of cell sorting. At this point in 2018, we hadn't studied it extensively. Hopefully, I'm going to convince you that we've done a very good job of studying this in our lab, and we'll cover that shortly. So there's a lot of best practices to minimize this stress. I think some of these will come out in this presentation and potentially in Rui's presentation after this. But a lot of these are kind of word of mouth, and they haven't been rigorously evaluated, other than the fact that, well, you get better viability. So this has always been kind of the initial screen for cell sorting. Did it kill the cells? The point I'm trying to make is, well, if it didn't kill the cells, maybe there are other things that could happen where the cell still is there and it still looks viable by a viability marker, but something's going on with the cells. And I think it's important to note that this exists. Sometimes you can't deal with it. And if you can't deal with it with a conventional cell sorter, there may be other purification technologies that you can use. And I'll talk about that. So in our case, since we coined the word, we kind of try to focus the attention towards 6 being something that is specific to the cell sorting process. Okay, so that's kind of how we try to discuss this. And in our lab, the goal is to understand what part of the cell sorting process causes 6, or what parts of the process are the cause of 6, and possibly there can be improvements made either on the user operator side or maybe on the vendor equipment side that will minimize this and make the sorted process, the sorted cells, a little bit happier after they get out of the machine. So again, this is that slide earlier that Mateus provided for the talk. And when we talk about 6, we're talking about everything here that's the sorting parameters, the pressure, the shearing, everything that happens with the sorter. The sample processing is hugely important, but we want to understand what part of the instrument process is causing this. I mean, we know, for instance, that you can, trying to get cells out of primary tissue can take a long time, and the cells may not be happy. And we're not looking at sorting results here. This is simply, what are you getting out of normal breast tissue when you try to disperse? So anytime we have these solid tissues that we have to break apart, we can have, pretty poor viability just going into the sorter in the first place. And, I think we can all agree that, putting a sample through the cell sorter that's starting out at 62% viability may not give you a very happy cell on the way out. So there are ways of, there's a number of best practices that could be used to try to deal with the situation, including viability markers, changing nozzle size, temperature, minimizing the time the sample's under pressure. This is one that I kind of like a lot, because we see people who are now trying to accommodate 15 ml tubes on the sample introduction side. Well, if you're putting 15 mLs of volume on a cell sorter on the introduction side, that sample is going to be under pressure. It's going to be sitting there for a long time. And it might not be, the cells that went through initially might be fine, but the cells at the end that are still sitting in the tube might already not be happy because of that pressurization and so forth. So, one thing to consider is, if you have to go through a lot of volume, is not to mount all of the material on the sorter at the same time. Just keep some of it either in the incubator or on ice. Media is an important factor. You can collect cells in media. You can use the media that the cells like to be in, or you can use high serum, or you could even use neat serum, just pure serum. You can buffer the media with heaps rather than using a CO2 buffer, and then the cells are not going to be in CO2. So these are all kind of common best practices, and I'm sure you may have others to add to this list. This is not everything, but there are things that we already kind of we discuss and we talk about, on the Purdue list and so forth for doing the best job possible. So, getting back to this notion of, some cells seem to have some problem after cell sorting. There have been a number of reviews and observations, and this slide is actually a little old because there's more that we can add here. But when we looked at these, two of these papers stood out in that they were both kind of in the same vein, and they were both saying something's going on with the cellular metabolome. And in discussion with Dr. Drew Jones, who's our metabolomics core director, we both came up with the idea that, let's validate this and let's see what's up. Drew runs a top tier metabolomics core. I'm very lucky to have him as a colleague and a co-author, because he really helped us with the study that I'm going to present. And it's interesting when you think about this central dogma, where people have looked for the effect of cell sorting. Things go from the genomic side down to the metabolomic side and ultimately to the biological phenotype. And, people have asked the question, and rightfully so, where, does the cell sorter, for instance, screw up the DNA because of the laser or the pressure or whatever? And it turns out that I think most of the papers say there's a minimal impact, if anything, on that end of the pathway here. But other parts should be evaluated. And we just went right down to the end here, and we said, look, metabolomics and the phenotype are like, the real-time result of the real-time kind of assay. So let's look there. That's kind of just confirmed what the other papers had outlined. So we felt, pretty good about going into this. And we just did a very simple experiment. We had some I think, I don't even remember if these were jerkettes. I think they were jerkettes. And we sorted them, and we had controls. Very straightforward experiment. And what we found through genomics was pretty interesting. Well, basically, we just confirmed what was in those other two papers. I'm not a metabolomics person, but you can see here that there's a ton of metabolites. And the condition to point out here in blue is where metabolites have been depleted. And this is the self-sorting, these are the self-sorting columns here. So the other columns are the pre-sort and the no-sort. Things are looking kind of happy. There's some depletion here, but look at this. This is just obvious as I was going to say obvious as hell, but I said it. But anyway, there is just, a lot of depletion going on. There are all these metabolites, and there's nothing that is less than blue, which means there's nothing that has not been depleted. If you look at a volcano plot, and I'll be honest, I'm not used to looking at these volcano plots, but I was told that's why I put this picture in the upper right-hand corner. They're usually symmetrical, meaning things are going down and going up. If you do a volcano plot of the metabolites in this situation, it's lopsided. Everything goes down. You don't have the component. I think there's one thing here that has gone up, but everything else goes down. And it turns out that these things can go down. Some of these metabolites have dropped 200 to 500 fold from what they were when the cell went in, before the cell went in. Things like D-glucose, citric acid. These things are being depleted. Everything seems to be depleted. So this was interesting. We confirmed what other people had already shown. We confirmed it in our own hands, but then we thought, well, let's investigate this a little bit further. And luckily, one thing that we have in our core at NYU is a cell sorter that I'm going to describe it here because many of you probably are not familiar with this. This is a product that used to be called the Synergy. It's now called the SY3200. Sadly, it's not available anymore, but this is a parallel cell sorter. This is two, I would call this two MoFlows in a box because they're both high-speed cell sorters. They're both independent. You can see here that there are two groupings of screens, basically two identical software packages are being run to control each one of these individual sorters. And I just wanted to also point out that I think they did a really great design on this machine because they didn't simply take a cell sorter and stick it in a box in a hood. Of course, we've got good biocontainment here. What they did was they took all of the heat components, everything, lasers and everything like that, and they put that in the back of the machine, and they worked with the hood manufacturer. This is a Baker hood, to actually put a hole in the machine in the back of the hood, and to only put the part that you need to handle under the containment, which is where you're putting the samples on. So it's kind of a great system. I'm really glad we had it because this really empowered this experiment that we did. We had a notion from other technology that there was, and specifically elutriation. I'm going to talk about that at the end of this talk. But in the context of centrifugal elutriation, we knew that if you put cells in an elutriator and the buffer that we used was PBS, we would see almost the same thing as a metabolic 6 phenotype. We'd see a depletion of a lot of metabolites, and we remedied that in the elutriator by using complete media as the buffer instead of PBS. So this was really just a hunch, just a notion, and we all agreed, let's just try this on the 3200. And this was where we came up with this crazy idea of using RPMI as sheath fluid. The cells were in RPMI, and we also had PBS sheath. This is just straight, clean PBS. This is no extra additives or anything. And this is complete RPMI. So it's got all the goodies in it, including serum. And to walk you through this, which is also in our publication, I've got the reference down here. We had some controls. The controls were freshly spun down cells that were immediately harvested. What this means is this sample here immediately was flash frozen for metabolomic assays. Then we also had some other controls. These were not sorted. This control was an aliquot of cells that was stored at room temperature for the duration of the experiment, and this is an aliquot that was kept in the incubator. So we had all these, and once the experiment were stopped, everything was flash frozen. Meanwhile, we sorted, and we had one of the sorters outfitted with RPMI sheath, and the other sorter, these are called HAPs, highly automated parallel sorters. We had the other one with PBS, and we ran the same sample, 2 different aliquots of the same sample. They look kind of close. You know, these are both independent flow cytometers, so they don't look exactly the same, but that's common for any. And you know, I think we all know that you can't get 2 cell sorters to look exactly the same as far as light scatter is concerned. Right? This is light scatter, and we only sorted on light scatter. And we did use a gate, so we tried to keep out the small debris and the large chunks. And we collected RPMI sheaths, samples here that had RPMI as sheath into PBS for collection fluid and into RPMI for collection fluid. And simultaneously on the other side, on HAPS2, we ran, which was outfitted with PBS sheath, we collected into RPMI and we collected into PBS. So this is kind of a busy slide, but hopefully it helped to walk that I walked you through it. And what we found was very interesting. We sorted a couple of different cell types. We sorted 3T3 cells and we sorted jerkets. So we sorted an adherent cell line and we sorted a non-adherent cell line. And what we found, again, I'm not used to looking at PCA plots. I'm sure many of you are, but here are our control conditions down here. And you can see that sorting with PBS and PBS, with sheath as PBS and then the collection fluid as PBS was very far apart from what we would see in the control settings. And even, you know, collecting an RPMI helped a little bit. It's moving us closer to where we want to be. But, you know, clearly when we go into this condition of RPMI, we're getting very close to having samples look a lot more like they did before they were sorted. So this was kind of exciting. We repeated this experiment many times, and I'll tell you, the metabolomics lab was really excited looking at this. They're not flow cytometrists, because they were under the impression that you could never do anything in the metabolomics world with cells that come off of the cell sorter. They've tried, they've always said the cells look terrible, and they were happy because they now thought that they now think we have a way of dealing with this. Of course, the way to deal with this is to use complete media as sheath. And I'm sure I'm not there, but I'm sure a lot of people in the audience are going, "Oh no, that sounds like crazy. That's crazy," and it is, really difficult. One thing that we also did in our paper was we looked at our data and came up with something called a 6 score, which is a number, and as the number approaches 1, those sorted cells are appearing closer to the controls in their metabolome. Bigger numbers are bad. And we came up with this because the assay that we were doing in the metabolomics lab to assay the metabolites was pretty major. And we wanted to look for a subset that might be easier for labs to adopt. And we found that if you look at amino acids, this is what we use for the 6 score. It's actually in the paper, the actual way to calculate this. It's in the supplemental materials. But this is a nice way, if you want to evaluate different conditions, you can do this using the 6 score, and you don't have to look at the entire metabolome. You can just look at the amino acids. So our kind of conclusion here is that between what we have shown and what others have shown, those other publications that I pointed out earlier, we believe that what we call the metabolomic 6 phenotype seems to be common and to droplet cell sorters, and it's cell type independent, which is kind of a nice thing, because if we're looking at specific cell functions, we may remedy it for that specific cell type in the cell sorting process, but it may not apply to another cell type. We find that this, we believe, we're open to other people's input on this, that this broad range of metabolite depletion is a common consequence of droplet cell sorting. You can collect into complete media, it's still there. I should note that we did not collect into pure calf serum. We have a lot more experiments to do along these lines. And if anybody wants to try that out, please do let me know how that works. But right now, what we've found is that if you use complete media as sheath, and we're going to call that CMAS, I'm all about the acronyms here. You can really minimize that, but it's highly impractical. But I can tell you it can be done because we did it for the study. I know when this paper initially came out, people were asking, well, does the 10% fetal calf serum cause a lot of foaming? We didn't experience that. I think if your waste catcher has an adequate vacuum on it, you're not going to have a problem. We didn't experience it. We've shown that this works in adherent and non-adherent cell lines. And we've also come to the conclusion that the RPMI is not replenishing the metabolites. Somehow the media as sheath appears to be protective. This is not my theory. This comes from the metabolomics folks, and I'm inclined to believe them. They really dive deeply into this data. So question is, does this phenotype, is it long lasting? We haven't looked at it. We haven't evaluated it. When you sort a cell, it's going to have it. We kind of accept that you're going to have this metabolomic 6 profile after the cell sorting process. Can the cells recover? Maybe they can. Probably they can. Maybe some of them will die in the process. Maybe some of them are activating some pathways that maybe weren't active when the cells went into the sorter in the first place. So these are questions we have to ask, and we have to do some further work. And it's something that to think about the long-lasting notion of this metabolic depletion. If you're going to use the cells immediately, if you're going to go right into a lysis buffer and you're going to extract nucleic acid, maybe it's not a big deal at all. But for all the other uses of cell sorting, this is something to think about. So again, I'm hopeful that Rui's going to be covering some of these things, but I just put them here anyway. That if sorted side streams are spraying, if you're seeing a lot of spray, that probably indicates something's up, like maybe the cells are too big, and maybe that's going to exacerbate the 6 phenotype. If the cell viability is really low at the onset, the cells are not in even the viable cells are not in a happy place. Maybe that's going to, maybe once you deplete down those cells of their metabolites, they're goners. You're not going to get them to grow anymore. So all of these things should be considered. CMAS, complete media as sheath, can help. It's impractical, but in our hands, it works. So again, one of the things that I'd like to point out is as people try to, if they have specific issues with their sorted cell product, where they're not functioning in some expected way, maybe look at the metabolites. Have a look at that and see if you can improve that as the first step. So what happens if you've gone through all this and your cells just don't like going through the droplet cell sorter? Well, then you can consider alternative technologies. I've got a couple of them here that I'll talk about briefly. You may have other favorite technologies that I didn't address here. If you did, sorry I missed them, and let me know. I'll put them on the slide next time. So we have a number of microfluidic technologies that have now hit the market. NanoSelect has the Wolf sorter on the left side, Miltenyi-Tito in the middle, and Nodexis has a system called the next one. These are all microfluidic devices. I should note that we have put the Tito through our assay, and we're still trying to review the results. I'm going to just hold tight with that because I don't have the results in front of me. But we have evaluated that system. I'm just going to point out that there's another microfluidic system which we have in our lab that we kind of like, which is this on-chip sort, which is not a lot of people haven't even heard of this system. It's a small company in Japan, and we have this system, and it's a very enabling system. We were able to get this study done that was very difficult to do. It was almost impossible to do using droplet cell sorting, and we got a very high-profile cell paper out with IPS-derived mice being in this blastocyst complementation assay, and that the only way we were able to get that to work was with that fluidic device. So that would have been a case where possibly even using media as sheath may not have helped. There's some new technology that's from a company called Acadium called buoyancy activated cell sorting, where cells are simply okay, I'm being told I'm over my time. And I can't turn off the clock. There we go. And where small balloons, microscopic microballoons, can float cells depending upon their specificity. The dep array is another sorting technology which is quite useful. It's very slow and cannot process a lot of material, but it can be helpful for small cells. This is used for circulating tumor cell diagnostic work at NYU. It uses dielectrophoresis to actually move cells from one of the chip wells to another, and then ultimately to be collected. And this technology, centrifugal lutriation, I talked about it earlier, and this is one that we have in our core. It's a purification technique, and actually this led us to the notion of using media as sheath. This can separate cells based on cell size, and it's been widely used for separating cells, cell populations from a heterogeneous population without a label. This is a label-free technology, so this is another one that could be used. And just to summarize, in the past, people had observed, issues with cell sorting as cell death. The cells died, and can't use them. That's a very specific phenotype, dead cell, right? But useless too in most cases. But now the notion is, there's more that we can look at. We know there are some cells that change that don't function properly. And sometimes we can make this we can deal with this by changing nozzle size or pressure, or maybe making a better prep up front. But this metabolic sorter induced cellular stress phenotype is something that we've observed in the cells that we've studied, and others have observed in other cell types. I would reference the papers that I showed earlier. So we believe that that's a common consequence. Basically, cells coming off of a regular cell sorter with typical conditions are severely depleted of metabolites. Now, if you believe that metabolic 6 always occurs, this may actually help us to understand some of the functional changes. Is the cell not functioning in X, Y, or Z kind of assay because the metabolites were gone, or most of them were gone, and they had to recover their metabolites. Until we learn more about what part of the cell sorting process contributes to this, push comes to shove. If you have to get something done, you can do it by putting complete media in this sheath fluid. Again, impractical, but it works. I just wanted to thank everybody. I'm going to thank Drew Jones and Rebecca Rose, who are co-authors on our paper. I'm going to thank everybody currently and in the past in the NYU Cytometry and Cell Sorting Laboratory, because they helped us to be able to get this work done. This is a very, very old picture of me working in Rochester in front of a slit scan flow cytometer, which is all that stuff in front of me. Just to point out, that right there is a disk storage, and I can't imagine that was holding more than 10 megabytes. But anyway, this is old school stuff. I even used to wear a lab coat, and I think a lot of you will snicker at that. So thank you, everybody, for your attention. I look forward to your questions. Be part of the roundtable. Thanks so much.
Thank you, Peter. Appreciate it. All right, now we're going to move on to the next talk, and then we're going to save the questions for the roundtable. All right, so our next speaker, our next and last speaker, is Dr. Rui Gardner. Rui's scientific career began just before graduating in biochemistry in 1997 as a trainee in mathematical biology. Didn't even know that existed. Oh, I'm kidding. All right, it's getting late in the conference, you can tell. All right, in 2004, he earned his PhD in biomedical sciences, for which the most work was carried out in the Department of Microbiology and Immunology at the University of Michigan, but also at the University of Southern California and the Gulbenkian Institute of Science in Portugal, followed by postdoctoral work on evaluating immune diversity estimation techniques, still at the Gulbenkian Institute, where in the end of 2006, Rui became the core manager of the flow cytometry facility. In 2016, Rui was hired to head the flow cytometry core facility at Memorial Sloan Kettering Cancer Center in New York. And since then, he has been involved in many professional activities, everywhere from ISAC in the core managers task force to obviously FlowTex organizing committee now. So without further ado, we're going to get Rui here. We're going to get your talk up. I forgot to get your talk up. Yeah, would you?
There we go. Okay. Yeah. Well, thank you very much for the kind of introduction that I wrote. It's really a pleasure to be here. I just want to say that I've been involved with FlowTex for now about 2 years, and it's been, a lot of people ask me, you're from New York, what are you doing? What are you doing here at FlowTex? But it's been such a pleasure to work with all of them. It all started with the beginning of the pandemic, and I won't go into too many details because then I will definitely go over time. But just to say that we started this working relationship, both with Memorial Sloan Kettering, the flow core there, that I run and FlowTex, and we had a lot of successful webinars and educational activities in the beginning of the pandemic and throughout the pandemic. So it's been again really a pleasure to be involved with such a great group of people. So the idea of my talk today is really to so this is a project or an attempt, an effort to actually bring to the community a series of guidelines that will help people perform cell sorting using best practices, but also to evaluate the performance of their sorters, and to also publish cell sorting data. So here I'm not going to go into all of those details, because actually Karen just showed a lot of these things, and all of the speakers today in this session have been addressing a lot of the potential aspects that we need to bring together and come up with a consensus. But today I'm just going to talk about cell sorter performance and impact of sorting on cells. Again, I actually won't go into too much detail here, but just to give you a general idea of and I think you've got that idea also from the other talks. There's still so much for us to discuss. We're still after so long, Peter mentioned, right? And I have a history of cell sorting slide here. I can talk about that. But for many years we've been sorting for decades, really, and we're still addressing fundamental issues that we need to come to consensus with. So I won't go into many details here, because Peter Lopez gave a fantastic overview of the history of cell sorting. What I want to point out here is that actually the history of flow cytometry began really with the development of the electrostatic cell sorter. And in fact, Max Fulwiler didn't have that much prior technology to work with. And so it was really impressive that he could come up with such an impressive platform that could sort cells. And I usually joke that if Max Fulwiler was a biologist, for sure, yeah, he wouldn't have developed a cell sorter, because as Peter mentioned, just the things that the cell is exposed to, it's really incredible. Any biologist would immediately say, no, this is not going to work, forget it. But fortunately for us and for all of the scientific community, the Mac just went on and developed the cell sorter. And for a few decades, actually not that much, but from maybe the 60s to the 90s, there was a lot of development, as Peter also mentioned. But actually, if you think of it, after the 90s, in terms of performance at least, cell sorters haven't really improved that much. And that speaks a lot about the initial development that Max Fulwiler did. Of course, in terms of optical performance, there have been huge developments, but we're talking about the performance of the sorter, right? How it isolates cells. The MoFlows that were developed in the 90s, the speed, the recovery, the purity, etc., is still the same performance that we see in the actually. I was going to say that we see in the new sorters. Actually, no, in the new sorters we're seeing less performance so far, but we hope that those things will be fixed. But again, speaks a lot about the way that the sorter was developed at first. So of course there are many limitations of droplet cell sorters, and just like Peter, I'm going to mention that I'm going to be talking about droplet cell sorters mostly, and obviously there are limitations. They require sheets. There are some sorters out there, not droplet based, that you can just dilute the cells or suspend the cells and run them through a sorter without having to dilute them. Of course, the sheet does require salts. This is very important to be able to charge the drops and be able to sort them. Peter mentioned also a lot, and actually everyone here mentioned a lot about the harshness of sorters on cells, and we'll talk a little bit about that. Of course, high pressure, electric charge, all these things, and the efficiency, and basically that's one of the reasons why the performance hasn't really improved since the 90s, is that the efficiency is limited by the natural laws of Poisson statistics, which is pretty good anyway, but it's still limited by that, and that's the difficulty in getting more efficiency there. So let me talk a little bit about cell sorter performance. So I'm talking about the instrument performance. And so, how this started, at least in my career, when I first started sorting, there was a lab in my institution, my former institution in Portugal, that they sorted B cells a lot, and typically they sorted B cells from the spleen. And they, they would bring the cells. Everything worked really well. Actually, one of the users was so happy once that came to me, and they said that, oh yeah, today it worked so well. I had 120% recovery, and I was really happy for that because it means that we actually give more than they bring. But obviously, the point was that it was working pretty well. And then a few people from their lab were actually sorting B cells from the peritoneal cavity. And every time they sorted, even on days that we sorted for their other colleagues, and things went well, very well, or even better than well. But with these cells from the peritoneal cavity, they were sorting B cells as well. They had maybe between 10 to 30% recovery. And we couldn't explain that. And obviously, I calibrated everything well at the time. I did operate cell sorters. And we tried to look at many other things. We started thinking, oh, maybe it's the centrifuge. You're washing the cells after you sort, and maybe you're losing a lot there because your colleagues just came here and we sorted everything perfectly. So the instrument has to be well calibrated. But the point was that I wasn't really 100% sure. I couldn't tell them, are you sure that the instrument is sorting the right drop at the right time, is charging the right drop at the right time? And actually, I couldn't. I mean, I didn't tell them that, but I couldn't be 100% sure. And that lingered in the back of my mind. And I started thinking of how the sorting process takes place, what's really happening there, and I started addressing the issue of recovery. And I won't go into details here because actually Karen showed a nice representation of how a droplet cell sorter works. But just to give you the idea of actually with this animation of how things are working, right? You're generating droplets, and then the cell, if you detect a target cell, the instrument will wait a certain period of time until that cell is at the breakoff, right? And then the instrument charges the stream. The droplet with the cell detaches from the stream, and then the instrument uncharges the stream. So only the droplet that contains the cell of interest is charged, right? So when it goes through the high voltage electric plates, it's going to be deflected into a collection tube, right? And I think this is important. That's why I like this animation, is that, you kind of see how, what's going on. Of course, this has to be a very extremely fast process that's taking place, right? Maybe you're doing this at 20,000 cells per second, maybe sorting at, at the rate of 1,000 or 2,000 cells per second. So it's a pretty fast process. Now, the important thing to understand is that actually droplet cell sorters are extremely precise at charging that drop. We usually think of, oh, if there's if there's a contamination, so if you don't have your collection sample is not pure, then the instrument wasn't being able to charge very fast, right? But that's not true. The instrument is really, really precise, and actually I usually say a droplet cell sorter should sort at a purity of 99% or more. If there's anything less, then there's something wrong either with the sorter or with the sample. May be difficult to overcome, but there's definitely something wrong because the sorter is supposed to be doing that very precisely. Now, of course, the timing may be wrong. And this is what we're going to see a little bit here, right? So how do we usually measure the sorter performance? Now, I highlight the word sorter because we're talking about the instrument, okay? So typically to know if our instrument is working well, we look at purity, right? Typically people are sorting for purity, and so it's important to check purity. Not saying it's that you shouldn't. You definitely should if you're sorting for purity. But as a measurement or a metric of the instrument's performance, it's actually really unreliable. So how many times did this happen for you guys that have sorted? You go to the sorter facility, you hand in the sample, you come back, and the person there says 99.7% purity. And then you go, wow, these guys are really good, right, Karen? They go to your facility. And well, I shouldn't say that, because then they go and back and count their cells, and you go, what happened here? Where are my cells? You know, purity was amazing, was impressive, right? So the issue here is when you start thinking of, and I've shown these slides actually at FlowTex in 2018 for different reasons, but I like this schematic because it kind of identifies, you know, the places where you can actually lose cells or compromise purity. And if you start looking at this, you know, cells can stick to the walls of the tube, or they can die while they're waiting to be sorted. They can get stuck or die, you know, throughout the sample line. And then, so this is going to compromise recovery. And then you can have things like, you know, you can have something wrong with the instrument in the electronics, or the timing is not correct, you know. So I call it drop charge delay and not drop delay, exactly because we're not delaying the drop, right? We're delaying the charging of the drop. And I think that's important to understand, again, conceptually how things work, which helps you then troubleshoot and get better results. But as I was saying, the point here is that, you know, there are some processes where you're going to compromise purity, but you also compromise recovery, because every time you're not sorting the right cell, you're losing it, right? If you sort the wrong cell, it's because you missed the right one. And so typically, what's going to happen is that on all these processes that you compromise purity, you also compromise recovery. But there are steps here that you compromise recovery and you don't compromise purity. And so everything kind of leads towards, and this is just a play of words here, where purity yields to recovery. And we did a lot of you know, studies on this, and we started realizing that, in fact, recovery is a much better metric to measure the performance of a sorter, whereas purity is actually not a good metric at all. One thing to remember is that if you, for instance, if you have a very diluted sample, and this happens a lot, if you have few cells, you know, most of your droplets are going to be empty. So if you're actually sorting the wrong drop, you know, you're one drop away from the right timing, what happens there is that most likely you're sorting just empty droplets. Once in a while, that cell, as Karen was mentioning, the sorting, the sort modes, sometimes that cell may actually move a bit slower and end up in the other drop that gets sorted. So you end up with a sample that's pretty pure, right? 99% or more pure, but you have very few cells because you've been sorting the wrong drop. So that's just one example. Then there are many examples where you actually have bad purity, but the instrument is actually performing quite well, because there are things that are happening after you've collected the sample and you've rerun them in the sorter. So like, for instance, internalization or downregulation of a certain receptor that you were using to sort, right, that you were identifying or labeling to sort. And, you know, if they're downregulated, when you run them again, you know, they'll have less signal and they fall out of that gate, and you think, okay, my purity is bad, maybe my instrument is not performing well, and it's not true. So this got us thinking about, okay, so if it's recovery, how do we measure recovery? And actually this starts, and Peter knows this, because this conversation started with DACO cytometry, where the idea was, why don't we collect the waste? Let's collect the waste. If any of our target cells are falling in the waste, that means we're losing them, right? Something is wrong with the timing, etc. And Andy Riddell had you know, a lot of collaborations with DACO cytometry and kept mentioning this. And so we put our heads together in a great collaboration, and we ended up developing this method called RMAX, which is basically collecting the waste stream. And as I said, trying to identify you know, what's being lost. The big step here was actually developing the math behind this, so that we could actually have a metric that would measure how our sorter was performing. And you know, I won't go into details. This is just to show that you know, the assumption here is anything that you sort will either fall into the sorted tube or into the center stream, right? So doing the math, you end up with a simplified, if you assume that purity is going to be high, and I'll show you why we assume that. If purity is going to be high, then actually this simplifies into a formula that just depends on, so the maximum recovery just depends on the ratio of the non-target and target particles in the original sample, and we always record our sample initially, right? So that's a given. And then all we have to do is to collect the center stream and look at the ratio of target and non-target in the center stream, and you get the maximum recovery in those conditions. So you know, we often do this in our cell sorters to measure to evaluate or to assess if our instrument is performing well, if it's charging the right drop at the right time. So basically, we run a set of beads with two colors. I'm sorry. And then we start sorting. As you can see, there's the sorted droplets here, and this is the waste stream that we're collecting. So we collect the waste stream, and then we rerun it. And again, if we had 100% recovery, we shouldn't see any of our target particles in the waste stream. You're always going to have some. I mean, again, the system is limited by Poisson statistics, and so it's never going to be 100%. You know, you're never going to have 100% recovery. But anything between maybe 85 and 95% is already pretty good. So you take those counts, right? The original and the center stream. You plug them in the formula, you get something like, in this case, 0.953, which means that in these conditions that we ran, we had a maximum recovery of 95.3%. Now, I don't know if for those of you who are familiar with our max, I don't know if you know where the idea of the name came from. The idea was that this is a way to evaluate what would be the maximum recovery that you could get, right? Because then, of course, cells are going to maybe die. They're going to be affected by, as Peter was mentioning, by the sorting process, but mostly by the post-processing. And so, you know, your recovery is always going to be less than this. But at least in terms of your instrument, this is a way to tell if your instrument is charging the right drop at the right time, right? So, and we've been, you know, advocating, maybe not as much as we should, but to implement this in as the best practices in your facility, or if you have an instrument in your lab. It's a simple process that takes maybe 10 minutes, not more than that. And once you get the hang of it and the details, again, it's pretty fast, and you can include this in your QC, in your instruments QC before you start sorting. And for instance, you can use this as you know, as a daily QC, just to make sure that your instrument is always charging the right drop at the right time before you start sorting. Or if you don't want to do that every time you start sorting, at least for some important sorts, like Karen actually was mentioning, you know, when you're doing a rare cell sort, it's difficult to measure the post-sort purity or recovery because you don't want to manipulate the sorted product, right? You don't want to lose even more cells. So for single cell sorting, or for precious samples, or for rare cells, or rare cell sorting, this is a good way to make sure that at least your instrument is performing well, right? And there's a lot of detail. I have here a link. We used to have it on the website of my former institute, but they took it down. But we placed it back on our website in the Memorial Sloan Kettering. So I encourage you to check this. We have videos of how to perform this. So pretty straightforward, and it's a great way to understand if your instrument is really performing well. We used our max actually then to start looking at different things in the sorter to try to understand the problems. This was actually kind of a serendipity. We were just comparing recoveries between sorters like the MoFlow and the Faxaria, and at the time there was a lot of talk about, and still is, a lot of talk about jet in air versus closed cuvette systems. And you know, there's been a lot of myths that have been propagated based on results that people have had looking at both these systems. To be fair, until very recently, the only sorter that really was a cuvette based was the Faxaria, and all the others were jet in air. Now, one thing to remember is when we say jet in air, is jet in air sensing. So the big difference there is just in the sensing, right? But then we're not taking into account all the differences. You know, if you compare a MoFlow with a Faxaria, in terms of fluidics, you know, all the valves, even the way it pressurizes the system is going to be different, right? So there can be a lot of things other than just the sensing that can affect the performance of an instrument, right? And I would say it probably affects more the optical performance rather than the sorting performance. But so we did these studies. We started looking, and this was what I was mentioning, and Karen already showed these plots where you can see that for the span of a drop, like from 36 to 37, your purity is actually incredibly good, right? You're not going to lose purity. But if you look at recovery, as soon as you start moving away, so if that timing is slightly off, your recovery starts dropping dramatically. And this is actually, all this was done in ideal conditions. So the curves are actually quite flat of recovery. But in real case scenarios, actually that curve starts tightening a lot. And so your loss of recovery is going to be way higher as soon as you move away from the right timing. Now, the thing is here, you can see that the ARIA, in fact, so what happened is that we started measuring using just random beads, and these beads were like 10 micron in size. And when we did the curve for the the recovery curve for the MoFlow, everything seemed to be quite right. So we were getting the top of that curve at the timing that we had measured, the drop charge delay. So everything seemed pretty well, but with the ARIA, it wasn't. It was actually shifted. And then we started thinking, could there be any issues with the fact that these beads are bigger than the beads that we're using to measure the timing? And so we started looking into that. And in fact, we did find out. So here you can see that for beads that are 10 micron in size compared to beads that are 1 micron in size, the ideal delay is actually shifted, right? So we were not sorting, this was measured with a 7.6 micron beads. And you can see that the 1 micron beads, the timing is not that off that compared to the 10 micron beads, whose optimal timing is actually quite offset. So of course we were losing if you're sorting cells that are 10 micron or larger, you would be losing way more cells just based on the instrument itself. Now we were intrigued with that, and actually again I was very fortunate when I moved to New York, where we had a lot of flow cores that were willing to look into this. And so we had several of these core facilities together with this Big Apple Flow Network that's organized by Svetlana Mazel at Rockefeller University. We put together a series of tests. We kind of standardized the tests that we were going to be doing. And we started measuring laser delays and the drop charge delays for these different instruments. And what we found was quite striking. All of the instruments actually performed pretty well, regardless of the size of the particles. But when we looked at certain areas, we saw that there was a dramatic difference. So here in blue, these are 7 micron beads. You can see that the peak of recovery is at the timing that we measure the drop charge delay, so pretty accurate. So we knew that the instrument was performing well. But when we tried to sort 15 or even 20 micron beads or 19 micron beads, we saw that timing was quite shift. But then some of the areas actually performed quite well, regardless of the size of the beads. And the only difference between them was that some had the rectangular flow cell and others had the square flow cell. So long story short, what we found was that, and BD is well aware of this, and actually they confirmed with us that yes, they knew that was a problem. So these instruments work very well if you're sorting, so with a rectangular flow cell, they work very well if you're sorting lymphocytes whose size are very similar to the beads that they're using for the drop charge delay. Now, for the case of a cancer center like ours, we're sorting typically cells that are 15, 20, 25 micron in size. And so we were losing a lot of cells here, and we were having a lot of problems, researchers coming back, what's going on? And it ended up being just a problem with the flawed design of the flow cell. So, we've been, we asked BD to change all our flow cells. They did change them for free, and actually the whole Big Apple Flow Network took advantage of that. So we had our flow cells changed, but I'm a bit surprised that not only BD is still selling these rectangular flow cells, but anyone that wants to change from rectangular to square, even if you have a service contract, they're charging several thousands of dollars, $25,000 25 or something like that, $25,000 just to replace, right? Something that is flawed. Imagine you buy a car, your brakes don't work, and you go back and the company says, or the dealer says, no, you have to pay to fix your brakes. It's been a bit disappointing, and that's why I'm bringing this up, because I thought that once we presented this data, that at least they would, they haven't done a recall. Think of it, the impact that this has had throughout these years. First, there's this myth that closed cuvette-based systems that are harsh on cells because we lose a lot of cells. Well, it's most likely based on this, right? And then you can imagine the lack of reproducibility based on depending on the size of the cells, the timing is going to be off. And so the impact that this has had in research in general, and if you think of the translational capabilities of research into medicine, the impact that this has had in people's lives, because DR is probably the most used cell sorter out there. So it would be great if BD could go back and at least stop selling these, and whoever wants to change them, this should be changed for free. And that is my pitch. So the one last topic, or the second topic that I wanted to address, is the impact of sorters on cells. And I know, so Peter spoke extensively about this, and he has his views. And actually, we haven't really spoken much, during this pandemic, and hopefully we'll have the opportunity here at the roundtable to actually discuss some of these things. But I have actually a slightly different view than Peter on the impact of sorters on cells. Or actually, I should say the impact of cell sorting on cells, because I don't think that the sorter is actually the most problematic issue. And here's just, he explained the study that they did by Camilla Ryan and the rest of the group and his at NYU. And so, just to summarize, what he mentioned was that, yes, in fact, it's interesting when you look at the metabolics, you do see that the cells that were sorted with sheath as PBS, they cluster away from the cells that were sorted with sheath as RPMI as sheath, and the controls, right? So this points to an impact of the sheath on the cells. Now, one of the things, and I would love to discuss this with Peter, one of the things that I notice here in this study is that the controls that they are comparing with were all cells resuspended in RPMI. And we know from metabolics that if you suspend cells in PBS, rather than RPMI, you're probably going to see a difference just there, right? So it's very difficult to conclude. So I'm not saying that the conclusions are wrong. What I'm saying is that the results are inconclusive, because if we had clustered this or compared this with cells that were not sorted but were resuspended in PBS, probably we would see the PBS sheet samples clustered together with the controls. For me, it's still, the door is still open, whether the cells are actually that affected by the sorter or not. And so another case was actually also work that great work that Peter, Andrew Box, Monica DeLay, and many others put together. So this was a very comprehensive study where they looked at actually several cell lines, or cells actually. So some that are robust, some that are not supposed to be that robust and should be, fragile and affected by a cell sorter. And they did compare it with a lot of different cell sorters. And the readouts were basically, they look at cell cycle, UV exposure, microarray analysis, and quantigene assay. And from all these results, the conclusions were cell sorters have little or no impact on the readouts that they look. Cell cycle, transcriptome, they didn't look at metabolics. And they also looked at UV laser exposure, and they also concluded that there was no impact of the lasers on the cells, which was quite nice. Now, if you look at it, at the actual results of the TISNI, one impressive thing that they did mention is that if you look at the results, most of these clusters here are from independent sites, right? So all of the the conditions, the readouts, etc., from a certain site will cluster together. So basically, what they concluded, and this is part of their conclusion, is that the data suggests that experimental noise from sources such as cell health or handling may have a larger influence than sorting or pressure, right? This is very interesting, and it's pointing to, wait a minute. So imagine this: you have a solid tissue, right? You're going to dissociate these cells. Cells have evolved in solid tissues to respond to cues from their neighboring cells to survive. As soon as you remove them from that environment, they don't have those cues any longer, and they enter apoptosis. So it's actually every time we dissociate, and we're mechanically enzymatically dissociating these cells. So a lot of harsh processes, even a centrifugation, is extremely harsh. And so we're doing all these things, and it's a race, to be able to keep these cells alive once we've dissociated them, right? So you can imagine that once they get to the sorter, they have to go through another process, which is harsh, but perhaps not even as close as harsh as dissociating these cells, or even if it just cells in a plate that you're trying to resuspend and trypsinizing them, even that is already an extremely harsh process, probably even harsher than the sorter itself. I can conclude that. We haven't done any systematic studies, but these studies here are indicating that. This was another also amazing study that was done by Graham and Joanne Lanigan, Ian Macara. The readout that they were looking at again was microarrays, and this what was available at the time for transcriptomics. And they did study a lot of different cells. Now the big conclusion here is, again, they at least from looking at microarrays, they demonstrated that FACS causes minimal disruptions to gene expression patterns, right? Again, no impact from the sorter. But one of the things they did, which I thought was very clever, is that they also looked at the transcriptome, so the gene expression patterns of the cells, right out of the of their physiological environment, right? So they had that initial control where they look. So this is the gene expression pattern of these cells, and now we're going to process them for sorting, right? And what they concluded is that actually they saw more differences between that that pattern of gene expression compared to before sorting and after sorting. So their conclusions, again, how cells are handled following isolation seem to be much more critical to obtaining accurate cell lineage gene expression data than one's choice of cell sorting technology. Again, they used a lot of, well, in this case, just two sorters, but again, here was the question of, again, a jet in air versus a closed cuvette sensing system, right? So again, this is all, so from all these studies, we haven't seen any evidence that there are that the cells are being that affected by cell sorting, but rather it's indicating that it may be during the sample processing that we're seeing real problems. This is another example. This was a test that we did with one of those microfluidic sorters. I won't say the name of the sorter just because this was just a one time, so we did it in 3 days. So it's I wouldn't say it's statistically meaningful. We only compared that instrument versus another instrument. So it wasn't really a fair comparison. We would have to look at many let's say many Faxarias, many Sonys, many Mofos, etc., to be able to actually come up with a good statistical readout. But the point here is that we also looked at in this case single cell RNA-seq as a readout. And what we can see here, at least in terms of recovery and viability, the first day actually seemed very promising for that technology, that microfluidic technology. But then you look at the other days, and the recovery was, typically the same, or if it was different for either one instrument or the other. So not conclusive at all that these microfluidic sorters, which sort at a much gentle pressure, much lower pressure, we're really having an impact on recovery. And if you look at viability, it's also pretty much the same in all of the instruments and compared to the unsorted, right? And when we looked at the gene expression, so the sequencing data, we saw that the patterns actually, they were different from the unsorted, but actually they were different both for the ARIA and also for the microfluidic sorter. And what we saw here, and again, this is very preliminary, so I don't want to jump to too many conclusions, but essentially with the ARIA, it actually selected more for immune cells, whereas the microfluidic sorter actually selected more towards the mesenchymal or at least you know, these type of tissues. So you know, what does that mean? We don't really know. Again, we didn't do much more studies on this. We just returned the microfluidic sorter, and we continued sorting with the ARIA. So you know, we could look into more depth into those results. And I won't go into details here. It's just, you know, the advantages of microfluidic cell sorting, the obvious ones. It's gentle, no aerosol formation. So in terms of biosafety, it's great. You have disposable microfluidic chips, small volumes, no loss of sample because it most of the times can be resorted, or it goes into a container where you can, where it's not diluted, and they're typically easy to operate. But so far the disadvantages is that the purity is not that good typically, and they're much slower, right, than the ARIA. Because what I didn't mention is that all these results, we sorted all these cells in 5 minutes in the ARIA, and with this microfluidic cell sort, it took an hour just to sort the same amount of cells, right? So, you know, if you want to, you know, if you really need a gentle sorter, you have to compromise in terms of speed, at least for now, right? Now, I'm not saying that the instrument doesn't, the sorter doesn't affect the cells. Obviously it does. What I'm trying to point here is that most likely most of the harshness, let's say the stress induced or stress induction that Peter was talking about, comes more from the sample preparation rather than the sorter. But there are some cells, depending on the cells, there are some cells that are really, really sensitive to sorting, and this was also serendipity. We were trying to sort these high 5, so these are insect cell lines, high 5 and SF9, and it was impressive. They were 95% viable. We would sort them, and we would have 5% viability. And it was, you know, anything that we did didn't improve much until actually it was the researchers that said, well, you know, we're culturing these in these huge, what do you call them? Right, exactly, bioreactors. And essentially they said that they used pluronic to, you know, to reduce the stress that these cells are subjected to. And so they asked me if that was a good idea to add pluronic. We did it. We added to the sample and to the PBS, and either one gave great results. And essentially, we went from 5% viability to 95% viability or more just by adding a little bit of pluronic in the sample. Just a curiosity, interestingly enough, if we added it to the sheet, we only had to use less pluronic in the sheet than if we added to the sample to be able to protect these cells. But the nice thing is that adding pluronic really did a great job. We actually tried that with platelets because that's one of the the applications that's very difficult to do with droplet cell sorting. But actually pluronic didn't protect them at all. They were still activated, and so if you want to do functional studies, it's very difficult to sort platelets on a cell sorter, on a droplet cell sorter, right? So in summary, what I mentioned here, in terms of cell sorter performance, is that we should not assume that the vendor instrument performs as stated, and this is one of the reasons. Well, not only because of that, but also because we were trying to understand where we were losing recovery, or how could we tell if the instrument was charging the right drop at the right time. And so we developed this method called RMAX that I would really recommend everyone to use if you want to understand if your sorter is really performing well. In terms of impact of sorting cells, so far what the data is showing is that there's little or no impact of cell sorters on cells. What's probably happening is that the cells are coming very fragile due to the sample preparation and handling of these cells. So it would be very interesting to start coming up with guidelines on, focusing mostly on sample preparation, but not only, of course. The instrument also has to be well set up, well configured for the specific application, for the specific tissue or cell type. So it would be great, and Kathy Daniels that spoke yesterday, and I have been thinking about this, and we want to try to get the involvement of the community to come up with these guidelines. Here we just call them my flow sort comparison. Basically, metrics that we would use to compare if we want to do comparisons between sorters, because all of this data is very hard to put together because everyone is doing it in a different way, right? But also to not only to these guidelines would serve also for the purpose of really understanding how to apply best practices in cell sorting, but also to what is the minimum information that we need to present when we publish, so that people can interpret correctly what we did with cell sorting, but also to be able to reproduce it correctly. And these are just some of the things that Kathy and I are coming up with in terms of the things that we need to address to be able to to come to a consensus in terms of how to perform cell sorting experiments. So this is a pitch. Actually, FlowTex was already actually it was Sarah that was proposing that we get together FlowTex, and we've done so many amazing things that I think this could be just one other amazing thing that we could do. Put our heads together and come up with these guidelines to help everyone just do better experiments in terms of cell sorting. And I'll finish here. Thank you.
Why don't we go ahead and get ready and get everyone in place for the roundtable. Hopefully we can get the camera changed. All right, we have the camera changed. Perfect. Hopefully. We lost Peter, I think.
Peter, if you're listening.
All right, so well, we're getting this set up. Could I have everyone take their badge and pass? And basically, I'm going to come up and down on pass it to your left, or bring it over to your left. I'm going to come and collect it for the raffle. We're going to try something different this year. Your left, your left. All right. So we'll collect these badges. Yes. No, I just need the badge. Pull them off. You can keep the lanyards. All right. All right. So we're going to do that. We'll collect all those. We're also going to do, so we're going to do, as soon as this roundtable is over, we'll do the raffle as well as in person, and we'll also do some online raffle as well. All right, so now that we've got that going on, why don't we go ahead? I believe we probably have questions in the chat channel, as well as we might have questions in the audience here. So if someone wants to raise their hand before Sarah gets to her computer, we can get you any questions for sorting. I will say significantly, I learned one thing today, if not anything, from all y'all. I mean, when it comes to sorting, y'all know a lot. And when it comes to talking about sorting, we have to give a hard deadline on when to stop. Because wow, that's a solid lesson. All right. So yeah, wow. All right. So first question is to Karen from Florenal Joseph. Is thresholding a good technique to use, especially if one is focusing on purity?
So it depends on what you're thresholding out. It can increase the rate at which you can sort, but it might reduce the purity of your sort product. And that would be if your whatever you're thresholding out, like red blood cells or something like that, will end up randomly distributed into your sort if you don't.
Yeah.
Is my mic on? Sorry. Is my mask on?
Yeah.
All right. So if you threshold out something like red blood cells, those would be randomly distributed into your sort product. So that might give you a lower yield. It's your downstream application from that that's going to determine whether that's a problem.
Mm-hmm. Right. Essentially, what the sorter doesn't see, it cannot avoid.
Yep.
So might end up in the collected juice.
Sure. So you need a threshold, and you just need to think about what it is for the experiment you're going to do.
Right. I have also to add to that, the other issue is, I mean, there's a balance, right? If it's too low because you want to avoid everything, then your efficiency starts dropping dramatically, right?
And you
so
can't finish your sort.
exactly,
It takes forever.
exactly. So there's a balance there, definitely.
All right. So we do have one. We do have one for Peter. So for the metabolomics study, where there is no sort cells being held, where were the no sort cells being held while the experimental cells were being sorted?
Well, if I remember correctly, we had a couple of them. There were cells that weren't sorted initially, so they went right into the assay, into the metabolomics assay. And then the other populations that weren't sorted were kept. One group was kept in the incubator, and one group was just kept on the bench.
All right, and there's
if I'm understanding the question right.
Yes, that's exactly what it was. Exactly. All right, so another question to you, Peter, is what extra cleaning of the sorter is needed when using CMOS instead of PBS? Media instead of PBS.
Well, I'd have to refer back to Camilla Ryan on that one because she did all of that hands-on stuff, and I know she got the system pretty clean. I don't remember the total time that was involved. I know she was not usually happy about the process.
I can imagine.
But I don't think there was much heroics there. I think it was just one thing that maybe it should be in the paper, but one thing for sure is that we did pre-filter everything and we took the sheath filters out of the system just to keep, because they're often a place where you can find a lot of residual. So it's in the paper, I'm sure, but everything was run without a sheath filter. So so that is like a preparatory step that can help in both of these, getting it back to normal. And to be quite honest, we've only done that process for an investigator once since then, and we're like I said, it's pretty impractical. If you could add something to the sheath fluid, like Rui was talking about, that would be helpful. But overall, it's a bit of a pain, and and it's simply an observation that we made that seems to work. And like we said, we believe there's something going on in a protective fashion. And like what Rui was saying, where you can use lower concentrations in the sheath fluid than for the insect cells, that may be pointing to the same thing, that there's something protective. And it has really not as much to do with what the samples are in or what it's really what's going on with the sorter. I mean, to be a little, upfront with where we're going with this. Yeah, I pointed out the droplet formation and that we used to sort 3 drops or whatever. It doesn't matter. you know, a lot of this is being driven by our metabolomics group, and they're amazing in both the fact that they're naive to the whole sorting process, but they're coming up with all of these places in the sorting process where they feel things are going on, you know, stuff that we take, you know, for granted every day. And our metabolomics core director believes that the sorter is an electroporator. He thinks it's an electroporation device, and an expensive one, and he even has gone as far to say that he thinks some of the cytosol is lost during the sorting process. So that's just to give you you know, a little insight into where we're going with future studies.
All right, and just a little follow up on this, but it sounds like we already heard the answer. Does anything grow in it afterwards if you weren't able to get it clean? I mean, like the sorters. I mean, were you guys able to get it clean enough where you weren't going to have contamination issues on your sorter post clean?
We'll put you know, again, we got to get to Camilla for the specifics. I think we typically did these experiments on a Friday, and we probably left the system sitting in something overnight. I don't want to say definitively what's going on. She would remember that, and maybe it's painful for her to remember that. But overall, we didn't see things growing afterwards. You know, again, I think taking out that sheath filter, any inline filter can help a bit, and we just did that. You know. Proactively, we just thought that was a good idea, but again, that means you got to sterile filter all of your fluids, which can be a pain too.
All right, I'm going to scan the audience real quick. Is there any question? There is a question from the audience. Any chance I can get someone to bring up? Okay, yeah, I'll repeat it. So the question is, has anyone looked at the oxidative stress of the cells going into the sort chamber, and if maybe someone has used an alternative gas for driving the sort?
That's something we certainly haven't done. I think a lot of sorters work off house air, and so there's not as much ability to do that easily. But I do have people who run oxphos assays after sorting and report good results. So I don't know that I believe it's an issue.
Yeah, I agree. We haven't tried that for sure, but yeah, I agree.
I know people have used nitrogen as to pressurize MoFlows in the past. If they didn't have the house air, but you know, we didn't really think about that question at that point. We're just happy to get that the MoFlow was working.
All right. Is there another? Nope. There's another audience question. Okay, this one's for Rui.
Right, that's a good.
Hold on, let me repeat it.
Oh yeah. So
would you repeat it, please?
Yeah. So the question is, if we're using a rectangular flow cell, right? If there are commercial products like, you know, bigger beads, right, that one could use to maybe measure the AccuDrop or something like that. So that's a very good point. We did start looking into that. Actually, the studies that we did, we asked Spherotech, and they were really kind enough to. They prepared custom beads, so AccuDrop beads with different sizes from 7 to 32 micron. So we had several of those beads to do this study, and we started thinking of that problem. But the issue is that when we're sorting, first just the distribution of sizes, even if it's a homogeneous cell line, the distribution of sizes is already quite big. So you're always going to be losing some of them, right? But then if you have PBMCs, you're sorting macrophages and lymphocytes at the same time, then you're stuck because you've measured that timing. So ideally, it would be great if the system could actually, you know, as the cell is going, measures the size and then would adjust the timing based on the size, right? But on the other hand, if all the other sorters perform well, where big cells or small cells don't accelerate or get delayed, so the answer to that would be, well, just fix the flow cell. Just come up with a flow cell where that doesn't happen, right? And that's what they did with the square flow cell. So BD is well aware that the rectangular flow cell is an issue. That's why they developed a new one.
All right, for a follow-up on that one, have you repeated the size and drop delay experiment on other sorters such as the Sony, the Bigfoot?
Yeah, so the Bigfoot, no, but the Sony we did. And actually we've been doing our max with the sorters that I've had in my lab. So from ARIA 1s to ARIA 2s and 3s, to the S6, the Sony SH800, the MoFlow Legacy, and then there are other people that did it with the Influx, the Astrios, and all of these worked pretty well, except for the ARIAs with the rectangular flow cell.
All right. We have another audience question. Okay, so I'll move on. So this is from Mariella Bolatti. So and I think this is addressed to everyone in a way. It's because it sounds like preparation, cell prep question. In addition to sheath fluid and RPI, we have seen that trypsin treatment of adherent cells affected the redox state of the cells. So could you guys comment on perhaps?
Yeah. Well, so again, and also answering a little bit the previous question, if we looked at the oxidative stress, and to be honest, any manipulation of the cells will impact the redox state of the cell, right? So, I mean, I'm generalizing it a lot, but I wouldn't say any, but most of the sample handling will affect in somewhat the cells in that respect. So that's why looking at metabolism is probably the most sensitive way of seeing if the cells are being affected or not. But they are being affected throughout the whole process of sorting, which includes the sample processing, includes the sorting, and then the post-sorting processing. So there's a lot of manipulation here that's affecting the cells, right? So I think that's why my pitch was more. I think we should actually focus way more on sample prep, which is probably causing the majority of the lack of reproducibility and rigor. And so we should start looking at what in the sample processing is really affecting the cells, fragilizing them or changing their metabolism, and even gene expression, because there seems to be some differences between the moment the cells are dissociated to the moment they get to the sorter, right? Rather than during the sorting process.
Really, I'm a little curious. I'm trying to also rationalize what you're talking about with what Peter talked about. I understand cell manipulation is going to just play hell with the cells going over the sorter. We all pretty much know this, anybody that runs a sorter. But Jurkats are buoyant T cells, very easily done. So then coming from what you're saying, is Peter simply centrifuging the Jurkat cells and putting them over, responsible for the complete metabolic depletion? How can we resolve these two?
Right. These are two independent processes, right? And each of them are affecting the cells, right? Of course, there are some cells that are going to be less or more affected, right, by the sample handling. Cells that are already in suspension, they're hardly handled, but even the centrifugation, I think, is already a harsh process. So it's really a question of for each cell type, and that's why it's so difficult to assess this, because a different cell type may react in a different way. The way that you handle the sample until it gets to the sorter will also affect how those cells will survive or not, or how they're going to be affected during the sorting process, right? And actually, that's the example I gave with those B cells, that coming from the peritoneal cavity, the sample processing was way harsher than when they were extracting them from the spleen, and possibly affecting how fragile they were when they went to the sorter, right? Because we were sorting with the same sorter in the same conditions, and with those B cells, we couldn't recover enough, right? So I think it's more that a question. So what Peter is looking at, he's comparing the cells that have been processed before sorting and after sorting, right? So it's just a small window of what's going on with handling the cells, with sorting the cells, I would say.
Yeah. So David, I mean, Rui's right. I mean, we're, like he said, one cell's going to be affected one way, the other cell's been affected the other way. And I've been going down this route for decades where, one cell does this, the other cell does that, and let's fix this and let's fix that. And you put something in, you put something out. And that's not our goal. Our goal is to find out what, just isolating the cell sorter, not the whole process. The whole process is going to have things on the front end and then the sorting process and then on the back end, like Rui said. And that's critical. That's an important question. But I've been trying to approach this either myself or with other people involved, and it just keeps on going around and around, trying to look at one cell type or the other. So we just said, let's focus on the sorter and figure out if, what's the sorter doing. And we think that, it doesn't matter what the cell is that you put on a cell sorter, you're going to lose metabolites. They're always going to come out with less metabolites than they went in. So the question earlier about, trypsinization and redox and so forth, that's how they're going in. And then the cell sorter is going to contribute the additional loss of metabolites. We feel pretty strongly that that's the case. And that's, we people to look at every other cell type in the world to tell us that. But, our feeling is that that's just what's going to happen. And, the quick fix is to use the complete media, but that's really not the answer. The answer is figuring out what part of that process is doing it. And if then that part of the process can be, re-engineered, can we come up with a better way of doing this and minimize that contribution to the whole process? That just takes one part of the equation and minimizes it a little bit. So that's really, again, Rui's right. We're just focusing on the cell sorter, the droplet cell sorter.
All right. Are there any audience questions? I think that was.
Just had one small thing to add, which was that I think that was a great conversation between Rui and Peter. Sorry, but it also strikes me that it was an important fact that the RNA expression changes were not observed. So, I mean, I think we're maybe seeing some changes in some things as a result of being on the cell sorter, but I don't think the take-home message to that is that cell sorting is going to mess up everything about your cells. They are still going to be functional and useful.
Right. Well, thank you, everyone, for that. That was a wonderful discussion. I think we're past time now, so we're going to move on and wrap this up. Thank you so much. Could we all give them a round of applause? Thank you, Peter.
Let me know if I won anything.
All right, I will. So let me just kind of for the streaming crowd at home and everything, we're gonna shake this up a little bit this year since it is hybrid. We're gonna raffle a couple off online, and although if you're in person and you win online, you can come down and you can claim it. And we're gonna get this all pulled up. We're also gonna raffle on in person as well. So if you're in person, you have every chance to win. The online people have a reduced chance, and I apologize.